Estamos esperando, né? Porque era para já tá aparecendo aí, né? Mas vai ser que nem o primeiro dia. Oi, Stefanela. Pô, ontem o pessoal da limpeza abriu. Quando cheguei aqui, 8h30, já estava aberto.
8h30, cadê o cara? Stefanel. Quem é Stefanel? Adriano Cortes.
algum som, alguma coisa só para ver como é que tá já.
Hello, good morning. Agora, hello, hello.
O Ed já tá
Hello, hello. Good morning. Just one moment, please. We are just finishing setting up the audio and the video infrastructure here, okay? Thank you, Giovanni, for the return. So, are you are hearing me well? Hello, hello, hello. Testing, testing, testing.
Era para estar saindo.
Áudio codec aqui.
Select
Use
speaker.
áudio codec até aqui. Speaker.
Isso aí. Era para estar saindo.
Hello, hello. Eles estão me ouvindo, tá? O microfone está funcionando pelo visto.
Eu vi um barulhinho de computador, não sei se foi aqui.
Hello, hello, hello. Testing. Tá saindo ali, ó. Tá
Mas eu preciso
movimentando.
que alguém fale para ver se.
Someone from the audience can speak a little bit just for us to test here.
Hello.
Just make some noise. Oh, thank you, Giovanni.
Tá saindo bem?
Hello. Yeah. Okay. Okay. Just one moment. Yeah. He will just slow a little bit. Okay. Can you speak again, please?
Hello. Good morning, everybody. Can you hear me well?
Yes. Okay. Fine. And you are hearing me well also?
I'm sorry?
Can you hear me?
Yes, perfectly.
Okay. Thank you very much.
Welcome.
Okay, can you hear me now? Can you hear me? Yes, yes. Okay, thank you, Anna. Thank you very much. Okay, good morning. Okay. Good morning. Hi, everyone. Can you hear me? Okay.
Peraí.
Tá. Okay, okay. Thank you very much. Sorry for the delay, so I have to hurry a little bit. But okay, this is about the game. Okay, this is our last day. Okay, so this is our last short course day. So I'm going to talk a little bit something that I'm started studying recently, and I'm so excited with this that I decided to share it with the students, with everyone that is watching this short course. So I'm not a biologist. I'm an applied mathematician. So this is why from an applied mathematician viewpoint. Okay, so and I'm going to talk about non-negative matrix factorizations for single cell Rna-Seq. Okay, data analysis. So let's start. Okay, first I like to say that this is our last day, as our Red mentioned. So I'd like to take the opportunity to thank all the speakers and the colleagues, Fabrício, Francisco, Marcelo, Stefanella, that together made the SysBio3 2022 happen. So thank you very much, all of them. Okay, a quick view, a very, very quick view, just one slide about single cell Rna-Seq. And I will try, actually, I'm not going to enter too much in the Rna-Seq, as I mentioned. This is something that I'm get used to. I'm starting recently, but I will give a brief introduction to what are the matrix factorizations applied in data analysis, specifically in biological data. Okay, so I'm going to make an analogy with a problem that is called LSA, this latent semantic analysis. So I guess we are going to try to catch it. So, and why a new matrix factorization? Okay, since we already have one that is most used by all of us, that's the PCA, the principal component analysis, that from the mathematical point of view is also the same thing as the single variable decomposition. Okay, so back to Rna-Seq and some reference. Okay, that I'm using. Okay, that's it. So here we have the whole, the main parts of the Rna-Seq pipeline. Okay, so we have the tissue. So we have a tissue sample. So we generate a suspension of this tissue. Oh, sorry. No, it's here, Francisco. It's here. Sorry, just one moment, please. You're having. Okay. muito sambando muito. Aí quando parece que vai funcionar, ele...
Exatamente.
Ai, caraca, bicho.
É.
Não vai ter tanta gente.
Muita ficção.
Okay, I will let you keep trying, and I will go in on here. Okay. Okay. So yeah. The point. Yeah. It's the audience here. Yeah. Okay. So I will keep going here. Okay. Beleza. Okay. Yeah. Okay. Yeah. Okay, so let's continue here. Okay, sorry for the interruption. That it stopped transmitting to the data show here, so we try to fix it. But okay, let's move on. So the single cell, so we have a cell suspension, and we have different technologies, but the most the usual one is one that's a microfluidic device that you have nano droplets, and inside this nano droplet you have ears, and you have like barcodes that somehow attach to each cell. So the things that you encapsulate each cell in this nano droplets, and somehow this is go through a process of replication by PCR, and then a sequencing and alignment, okay? And then you end up with something that is our data matrix. So this is the count matrix, okay? This one here, okay? Okay, and there are a lot of other steps here that I will not mention too much. This reference here down, it's very nice, very accessible. I recommend for you to take a look. He discussed every step here, very interesting. So, but what we want to talk a little bit today is what we do with this data matrix. Okay, there's some quality control checks that must have been done, but I will accept that we already have it. Okay. That somehow is the count matrix, that is a matrix that is genes, okay, that is genes here, and cells or samples, okay, here. And we are going to, this is what we call the downstream analysis. So this is where data analysis come into play, mainly those techniques that is dimensionality reduction here and clustering also, okay? So, okay, this is also another point of view that I took from this paper here. Actually, the name of the paper is very interesting. That is Enter the Matrix. So somehow Enter the Matrix, Factorization uncovers knowledge from omics. For sure, the title of the paper makes reference to the Matrix movie. Okay, and so it's very interesting. But okay, again, after all the pre-processing step, okay, I did not mention here, so we can say that all these steps here are the pre-processing, okay, steps, and then we also end up with this data matrix here that is genes per cells, and somehow we have the concentrations of the measured genes in each of those cells, okay? Okay. So let's move on to this analogy problem that I mentioned before. So we are going to look for this example from the text mining area. Actually, this is not a recent technique. It's we have things that are more recent. But okay, let's think about this model that we call the bag of words model, okay? And we want to promote the topic recovery and the document classification based on a matrix factorization of what we call the term document frequency data. So we have a corpus. We have a corpus of documents, okay? So we have documents, so this is our samples, okay? And we have the words. We have a vocabulary, okay? So we have a bag of words. So in the documents, so this is why I call it bag of words, because we are not seeing the documents and the words in terms of their relative positions. We are only seeing them as counting them. So we have, let's say, one word here, word one appears, let's say, 2 times in document 1, 0 times in document 2, 1 time in document 3, and so on, okay? And we end up with this data matrix. So start thinking about this an analogy. So we have genes in the rows, and we have samples in the columns, okay? So, so we want to do a matrix factorization. So what is a matrix factorization? It's somehow to come up with a product of 2 matrices that hereafter are going to use W and H, big W and big H, and WH, okay? And this is an approximation, okay? So we have and generally we ask this to have M rows and N columns, and here we are going to have M columns and K rows, sorry, M rows and K columns, and again, K rows and N columns. So once again, so the first matrix we have M rows, K columns, K rows, N columns. Okay, that's it. With K much less than the minimum of M and N, okay? So that's what we call the rank. This matrix has something particular in it, that their entries are non-negative. And this is why an analogy also when we think about the data matrix from Rna-Seq, because when we think about the count matrix, something that comes before normalization or log transformation techniques, but we have a counter matrix that take into account the number of the measure of the genes, okay? So, and what are the topics, okay? The topics are the set of words that are found simultaneously in different documents, okay? Let's remember a lesson that Gilbert Strang, since a long time, is teaching us about how to really think about matrix multiplication, multiplication, matrix product. So remember that the first column here, okay, is the linear combination of those columns here, the columns of W, with the coefficients coming from here. Oh, sorry. Coming from here, okay? So these coefficients will multiply here, this coefficient will multiply here, and this coefficient will multiply here, and the linear combination we're going to give us the first column. So we somehow have a set of column vectors, set of linear independent column vectors that are capable of reconstructing all the documents, okay, once we have the right coefficients. So, and this is what we mean by classification, because we have, okay, the coefficients here, we can also call the weights. So the weights of each topic is capable of reconstructing the documents, okay? One way that LSA obtain this is by using an approximate solution that is the singular value decomposition, the PCA, okay? That we know that can be cast as an optimization problem. Is this optimization problem here, okay? This optimization problem here, that is to minimize a measure of error. Here we are using the square of the Frobenius norm, and we know by the Eckart-Young-Mirsky theorem that the solution of this problem is possible. Actually, something that I have to mention later on, this is a non-convex problem, as we have for the non-negative matrix neutralization, but we have a simple solution. Actually, is the SVD, something that we are using since the 70s by all the work of Gene Golub and all those brilliant guys from numerical linear algebra. Okay. So the solution on here. So W is what we have from the matrix U, okay, from the what we call the left singular values, sorry, the left the left singular vectors, sorry, okay. Sigma is the singular values matrix, and those are the right singular vectors, okay? So we can come up with W as a truncation of the U matrix, taking K columns. So we have U, it's a complete matrix. Here it's M by M matrix, but we take only a part of it, let's say K columns. That's what we call this the matrix U, and this is what we call the matrix UK, okay? Okay, so I have here something that it's a scikit. So I'd like to start sharing here my own screen. Okay, let's share here desktop one. Okay, I want to share this one here, okay? So can you see my screen now? Yes, okay. Oh, how do I access my, how do I remove this one? Okay, okay, I will let like this. So this is an example from scikit-learn, okay? I do not have too much time now to enter in too much details of this example, but this is what we've been talking about here. This is the text. So this is a very known data set. This is the 20 new news groups data sets. It's a bunch of text from 20 different topics. That is the beginning from the internet, when news group was something that we used to communicate with each other. So I took here, I guess, 6 topics. One is atheism, one is religion, computer graphics, scientific space, something about motorcycle, and something about computers and math hardware, okay? So I'm taking 1,000 documents and a vocabulary of 2,000 words, and K, our rank here, K is 20, okay? And I'm going to show you somehow the ranking of the topics of the 20 top words, okay, and each topic. So I have a lot of things here. Okay, let me start running here, all the cells. Okay, this is very quick, so I don't have too much time. Okay, so this is everything I'm fetching here, all the data. So I'm only taking the categories that I mentioned before. So not taking the 20, only 6. And here, Scikit-learn has all the infrastructure for you to build this data matrix that I mentioned before, that are the term frequency document matrix, okay? So, okay, let's run the cell here. So I have what we call this TF, the term frequency data, okay? And I will do the SVD, okay? And I will do the SVD, so it's very, very, very fast. And I went up here, okay? So this is the what the topics that somehow SVD, the matrix factorization, somehow this black box, this Oracle magic box came up with us. So the first topic, it's this bar here is the weight, is the weight. So somehow it's appear to be related with religion because it come up with Jesus, Matthew, but then we come up with processing, software, analysis. So this is really, really a funny exercise because somehow you see that it mass something. So here also we think, I guess this is now, it's really about religion because Jesus, Matthew, prophecy, Messiah, Isaiah, David, God. And so this second one, we think about, okay, space, Mars, lunar, maybe this is related to space, okay. And then we end up with motorcycle. So motorcycle, it's okay, we can go from motorcycle to the space. So somehow he put motorcycle as a word related with this. So you have all here. So okay, again, put Judas with space and Matthew with telescope. So somehow it's messing the talks. Okay, so okay, let's stop here. The next one we are going to use it the non-negative matrix factorization. So let me return sharing here my tablet. Okay, so again, I have here the tablet. Okay. Okay. So this is, okay, didn't change. Okay, this is another example that we clear see things going on. That is called the Eigen Phases. Okay, so we have a bunch of image of face, very aligned, cropped, even other than that, we're going to have some troubles, but okay, they are really cropped to somehow match the position of the eyes, nose, the mouth of those people. And we vectorize each image. So we have a matrix, the image, each photo, it's a matrix, two-dimensional matrix. Somehow we vectorize it and we put them as columns of the data matrix. So now we have our features, let's say our features are the pixels positions and the level of light intensity, okay? And we have this bunch of faces. So we can also do the SVD. So we can also have the SVD and come up with the W. Okay, this is not equal, this is an approximation. So W and H. So actually, U and H is the same here. Sorry, H not W. W and U is the same here. So each column is somehow is a ghost face. That's what we call the Eigen Phase that you can, as I mentioned before, using the weights, taking a linear combination to reconstruct an approximation of each faces in your set of faces. Okay. And this is taken from this paper here, that this Lee and Swan, that is learning the parts of objects by non-negative matrix factorization. Okay, it came up in Nature in 1999. So it was it somehow positive matrix factorization came before that with people from other areas, but somehow people nowadays have a consensus that this was the really first paper that took this as we know today, that used this as we know today. So this is the same thing we did before. So we have the PCA, and here we have the Eigen Phases, and here we have the weights. And what I want to mention here before, the same before here, we just return here. This data matrix here is also made up by non-negative entries, okay? So those are non-negative entries. So the level, the intensity of its pixel is in the grayscale, and also RGB. So it's one form, it's from 0 to 255. So it's non-negative numbers, okay? But when you compute the Eigen Phases, you can see here by the red values, you end up with pixels that are negative somehow, that has a negative value. And also on the weights, you have negative values. So you reconstruct the faces, but using something that people used to say that not very interpretable, in the sense that you have faces, but with negative values, okay? So this is what I have here. So why a new matrix factorization? So this is what I mentioned before. This is now it's a consensus that is the seminal work of non-negative matrix factorization as we know nowadays. It's, as I mentioned, it came in Nature in 1999. Okay. And when we take the same problem, the eigenphase problem, and we do the matrix factorization, so we have, again, the data X, that our data matrix are the faces, okay? And we take the factorization, okay? Now we do not have any more negative, because this is something that I'm going to mention later in the next slide, but I have to mention here, we ask for the entries of W to be non-negative and the matrix of H also to be non-negative, okay? And so what we can call the eigenphases here, as you can see from the image here, it's learned somehow the localized features. So we can see here that we have something that is similar to a mouse. We have something here that's similar to eyes. We have here that's something that's similar to noses. So somehow we split in parts the objects, okay? And also the weights are non-negative, okay? So this is the original image, and this is the reconstruction by the parts learned by this matrix factorization, okay? Okay, I'm not going to enter into much detail, but this is the non-negative matrix factorization problem. So we have to minimize a measure, an error measure, okay? So we have, again, a data matrix that's a non-negative matrix. We have a distance measure, okay, between the two matrices, and we have to find W and H, okay, so that this distance is minimized, okay? Non-negative matrix factorization actually is what we call a dimensionality, a linear dimensionality reduction technique. And we know since PCA that this is a very powerful technique, okay? And also we can see it in an equivalent way, what we call low rank matrix factorization, because remember that W is M by K, so it's at most rank K, or what we call rank K, or rank K, and this is K by N. Okay, so this low rank matrix approximation actually a huge, a big amount of work and papers nowadays addressing this kind of thing, because this is the basis of a lot of data analysis algorithms. Okay. Just some comments about, okay, you can ask, okay, but what is the error measure? Okay, this is an important choice because it is somehow related to the statistical model for the noise, for the error. Okay, we use the same thing that we used before, the squared Frobenius norm. This is related to independent, identically distributed Gaussian noise. So it's the same thing as PCA, okay? But as we see before, this is somehow an interpretable factorization, okay? Because as we see from the phases example, the Eigen Phase example, W is capable of somehow learning and come up with part-based representations, okay? And the non-negative FH, okay, and also imposing the non-negative entries of H, we end up with basis coefficients that we do not have cancellation, because this is an additive model. So we can only add. So this is the difference between the PCA, because PCA, the Eigen Phases, we have positive and we have change of sign. And also on the coefficient, we have change of sign. So somehow they cancel together to reconstruct an approximation of our original image that is made from positive, for non-negative entries. Okay, so that's the thing. So learning by parts, it somehow began interpretability, and this is why a lot of people nowadays is looking for this matrix factorization, and we have really a lot of variants. A lot of variants came up since the last decade, and a lot of them from computational biology literature. So that's why we have to look at those. We have to start looking close at those things. So the challenge, the mathematical challenge are, this is a no-convex problem, and people used to say that's a hard thing to solve, and actually it is somehow, but we have a lot of theoretical and algorithmic progress recently. We have an issue that is identifiability, okay, that come up with the thing that we do not have uniqueness of solution, okay, but there are, as I mentioned before, a bunch of variants that we can impose more constraints on W and H that can fix this issue. Okay, you can put as for sparseness and orthogonalities. So this is somehow fix those problems that you can see this in the literature now. Okay, so let's return very quickly to our problem in the. So now I'm going to do the same thing. As you can see here, I'm using the scikit-learn just to show here. Scikit-learn already have the non-negative matrix factorization. Actually, the loss function here, the error function, it's an improved one with a bunch of terms here that somehow take into account all those some of those variants that I mentioned before. Okay, but here it's the plain one with the Frobenius norm. Okay, so And here we have, okay? So those are the ranking of the topics by the non-negative matrix factorization. And somehow it seems that it's ranked things. So you can see in the top one, it's image, data, process, software, analysis. Topic 2 is Jesus, Matthew, prophecy, people, said Messiah. And topic 4, it's probe, Mars, lunar, moon surface probe. So you can keep going on. You see that all the words somehow related to space topic. And okay, this I guess the top 8 is from computer graphic for sure, because it's a P2, P3, P1. Those I guess are points from a triangle, radios, P4. If you're taking like a quad mesh, that's something that people use a lot in computer graphics. So square root, sine, cosine. Okay, this is very, very, very fast, but I just want you to take an opportunity to do this experiment in your place, taking this example there. Actually, I cleaned it up a little bit, the example. The example do a lot of different, compare a lot of different matrix factorizations, okay? So let's quickly return here because I'm running out of time. Okay, so let's return from topics to metagenes. So this is actually also was the first example that people use non-negative matrix factorization. That is came in PNAS in 2004, and the metagenes and molecular pattern discovery using matrix factorization. This is not Rna-Seq. This is actually the older cousin of Rna-Seq. That is micro RNA assay. But okay, it's again the game is the same. Take your data matrix, do a factorization, and try to extract those information. So now we do not have like topics, but what we can interpret like is metagenes. Okay, and this is an example. Okay, and you can see from the matrix, actually from the matrix H here. Okay, that somehow it was possible to see here 2 classes. It could be used for cell type identification. Okay, so and you can see somehow you have this this analogy. Okay, so I expect here that the non-negative matrix factorization could come up with those vector columns here that are somehow more separate better the influence of each genes or the set of genes in each cell type. So the set of genes that are activated in different cell types, okay? And also process also, okay? Not only what people use is saying that the identity program, okay? So, okay, this is again from, this is a more broad perspective, this paper here, the entry in the matrix, factorization uncovered knowledge, because it said that, I'm not saying that NMF is very superior to PCA, this is not my message here, but I'm saying that we have to start looking also at different matrix factorization, and what this paper claims that you have to look for, you have to do the study of your data matrix with all of them, because each one can say something to you. So he used this paper. It used nomenclature here that the W it's called amplitude matrix, okay, and H what we call the pattern matrix. And as I mentioned before, each matrix can be used somehow for different tasks that are become common in the data analysis of biological data. Okay, so I mentioned before this here, that is the subtype discoveries. Okay. Okay, so I have 2 minutes now, but as I mentioned before, this is not start and end class, because I could not make an example here, a Python, a Jupyter notebook with an example here, because we don't have time. But I leave you with a homework, okay, that I'm also doing by myself, okay, that is taking this paper. I guess this paper is from 2019. It came up 2019. Actually, it's fresh, okay. And the title is Identifying Genes Expression Programs of Cell Type Identity and Cellular Activity with Single-Cell Rna-Seq. Okay, they use matrix factorization. Actually, they come up with an idea, what they call the consensus matrix factorization. This is an idea that was already present in the paper of the metagenes in the PANS in 2004. Okay, that's somehow to you run a bunch of factorization with different initial, because they are done. I did not mention this before, but the algorithms to solve are iterative algorithms. So you have to start with a W0 and a H0 matrix. So, and then we iterate. But as I mentioned before, you have the identifiability issue. So you can start with different seeds, with different initial matrix, and have different runs, and then take an average, take a mean of those things, and come up with what they call the consensus matrix. Okay, that's something actually that came from hierarchical and clustering algorithms, okay, that are also like k-means, non-convex problem. You have local minima, so if you start with different clusters, with different centroids, you can end up with different clusters. So this is also, you can do in the consensus matrix, you can also do in clusters. So this is actually a ingenious idea that you can also use in non-negative matrix factorization. And in the first author, the Dylan Keutler, you have, if you type this title in Google, it's going to appear the paper, and also the link to the first author GitHub. And there you have a Python notebook that you can follow it, and you can do all the analysis. So it's a reproducible experiment you can do with this paper. So thank you very much. Those are the reference that I used, but I put most of them during the presentation, the slides. Okay, so once again, thank you very much for being here in our last day. I hope you enjoy our next talk. So I'm 1 minute late. Thank you very much.
Deixa eu ver. Não.
Hi everyone. Okay, so just let's wait a little bit. The next speaker will join soon. Okay.
você está aqui. Oi, Cíncia, tudo bem? Oh,
Tudo bem, tudo bom, você?
sim. Esta modalidade, eu abro a sala de aula e no celular. É onde, então, o congresso está no celular. As pessoas trabalham aqui, onde estavam dando aula, alguém estava falando na minha bolsa. Eu tinha esquecido. Ai, gente, esse mundo moderno é meio louco. Então, aí vamos começar em 5 minutinhos, tá bom? Daqui a 5 minutos eu volto aqui, tá bom? Queria dar um oi. Ok, daqui a 5 minutos.
Tá bom.
Hello, everyone. So we are going to start just in 1 minute. Wait, there are people here finishing the coffee break. Just 1 second. So, okay, I think we can start. So it's a great pleasure to introduce Cincia de Oliveira Laje Ferreira. That is actually is also a colleague, is a collaborator. Was a real pleasure to get to know her over a year ago. And Cincia, she did her her undergraduate at UERJ. She did a master at PUC, and then she went to France, in Toulouse, where she did her PhD. And she's currently at USP San Carlos, where she works on problems related computational and geometrical methods, applied mostly to biological problems. So Cincia, please, it's a pleasure to have you.
Thank you very much, Stefanella. I would like to thank you also the organizers to be here. In fact, it's a very nice conference. I'm very happy to this opportunity. So I'd like to thank also my colleagues. You are part of Rodrigo, George, and Adenilson. We are part of a group, ACD Bio group. It's a biological computational analysis. We are interested in this kind of data, especially in cancer data. So today I will speak about topological characterization of cancer driver genes using Rectome super pathway networks. It's a big title, so I will cut this title in 3 parts. So I will speak a little bit about cancer driver genes, about topological characterization, and what are Rectome pathway networks. So I will start by cancer driver genes. So cancer, you know that's a molecular disease caused by genetic mutations that leads to cells uncontrolled growth and division. So you know that is a complex disease. So in here you are interested in driver mutation, the mutations that are responsible to cancer. So you know that tumors undergo a large number of mutations, but only a small portion contributes to oncogenesis and tumor progression. So here you are so interested in this kind of genes, this kind of mutations. And you have a lot of databases concerning these mutations, these genes. Here we have Intogene. This is a picture showing this kind of genes. The symbol, the size of the gene here, it's relative to the count of samples with mutation in that gene. So you took this database and also NCG database, it's another driver database. You considered the union of these 2 sets of genes to do our work. Also, we use the Rectome super pathway networks. What's a pathway? So in very small explanation, Rectome is a pathway database, so we use this database concerning pathways. Pathways are a set of genes that interact to perform a specific biological function. So it's a set of genes only. It's not a network yet, but you can model as a network after I will explain how you do that. So Rectome has more than 1,800 pathways, and they consider the union of these pathways in only 26 super pathways. So you consider these ones 26 super pathways because they are bigger though. So if you model using networks is more interesting than these others here that are small sets. So the examples of super pathways you can have cell cycle, circadian clock, programmed cell death. It's a very interesting pathway. You are very surprised with this pathway here. Our preferred pathway. Reproduction is also a super pathway. So, for example, Rectome has the pathway of reproduction. So you have this 81 genes associated to this function. So the set is here. So our genes, isolated genes, for instance. And among these genes, you have 14 genes that are drivers, no drivers. This set of drivers, it change with frequency because it's not all new days you have new drivers found. So here we have 14 drives in this pathway. So since these genes interact with each other, you can model them using a network. So for example, here also is the network representation of the pathway reproduction. So we can see here all the set of genes, they interact to have here a group, a set of genes separated from the others. So this network has topology. We are interested in this kind of study. Here it's the same network at reproduction pathway. So we put in red here the driver genes, only to look where they are. So we have 81 nodes, about 800 links, and drivers, 14 drivers, as we saw before. Another example here is the programmed cell death. It's another pathway. You have 200 nodes, genes, about 2,000 links, and 27 drivers in red. They are here represented in red. In red, you have this structure here, nice structure, and you can observe that it's the topology of this pathway is different from the other. So it's this kind of analysis you are interested in, and to observe the whole of the driver genes, the driver nodes. So to illustrate the topological characterization you do, I will give also a simple example. Here you have a sample network. It's not a path. It's only to explain what we do with this. So we have this network here. You can consider measures of centrality. So we have some of them, but here we consider 4 measures. So we consider degree. What is the degree? It's very simple. It's the number of neighbors a node, in this case, a gene has. So here we have 1, 2, 3, 4, 5, 6, 7, 8 neighbors. So this node, consider this measure, he is a central node. Also, this one is central because also he has 80 neighbors. And the others, the yellow ones, are not central because they have not a lot of neighbors. But when you change the measure, the central measure, you can change also the whole of the nodes. So here you have closeness centrality. It's a centrality that measures how close one node is from the others in the network. So these nodes, 3 nodes here, they are closer than other zones because I can go to all parts of the network from them quickly with a few steps. And this one, for example, is far from the other. So you have no, it's not a central node considered this measure. You have also clustering, consider also clustering coefficients. It's another measure. It measures the interconnectivity between neighbors. So this node here that you have a very big degree, he has clustering 0 because his neighbors are not interconnected. There is no links between his nodes. This node here, he has a big closeness because it has one clustering. Clustering is between 0 and 1. He has one clustering because all your neighbors only 2, but they are interconnected. So you change here the whole, again, the whole of the nodes. The last one we consider is between the centrality. What it means? Between centrality concentrates the flow of information. So many pathways, many ways pass through this node. So if you are some information starting from all nodes, so they will pass through these nodes. So they are very important considering this measure. So in this way, you are interested to investigate the hypothesis that driver genes are topologically different from other genes in the same pathway. So you are interested to look the central role of drivers. To do this, so we considered the Rectomic functional interaction. This is a network of protein interaction. It has about 1,400 nodes. We consider also the Rectomic pathways, that is the list, the set of pathways of genes, and we considered also subnetworks. So you get this set here, you look at this network, and we take it the subnetworks consider this set of genes. So we construct the super pathway networks, and after with the driver database, we consider here the driver nodes. So there is a simple model. To do this analysis, I talked to you that we have 26 super pathways, but here you are interested in 2 drivers, so we consider this 7, 1, 2, 3, 4, 5, 6, 7, 8, no, 7, and the last one is the union of all super pathways. So we consider this 6 super pathways that have the most percentile of drivers. So, for example, here, reproduction, we have about 100 nodes in the biggest component, connected component. It represents this percentage of the all genes. This is the total genes, and here it's the biggest component compound, so about 7% here. So this set here, we have 14 driver genes. This is 17% of the network. So you take the biggest percentile here, and we consider also the consensus, the union of the all 26 pathways. Then you have 11,000 nodes. The biggest component compound have 90,000 nodes, and you have about 7% of driver genes in this biggest component compound. So we did this analysis here. You calculated the distribution of the centrality measures I talked before. So here you have the distribution of degree, clustering coefficient, between the centrality and closeness. And here we have the DNA repair, programmed cell death, reproduction, and consensus of all super pathways. And the box here, the blue box, are non-drivers genes. So the degree of non-drivers genes, the clustering of non-driver genes, and so on. In red, you have driver genes. So here you have the degree of driver genes, DNA repair, and so on. And in yellow, you did a random sample of the network in order to observe if the behavior of driver genes is not random, for example. For example, in DNA repair, in fact, if you look the distribution of driver genes comparing to random samples, here we have one random sample with the same length of drivers. Here we have 10 random samples with the same size of the driver genes, and here we have 100 samples with the same size of driver genes. In fact, the DNA repair is a very difficult pathway to analyze for the moment, because you observe that all the measures plays the distribution plays the same role as random samples, considering if you compare with driver distribution, driver genes. But programmed cell death has an interesting behavior, because here we have the degree of drivers, non-drivers, they are different, and it's also different from the random samples. So we have this comportment to degree, also clustering. You have a small clustering comparing to non-drivers, and it's different to handle samples, and also between us is quite different to non-drivers and handle and also to handle, and also closing centrality. So it's a very nice pathway. Reproduction also, you can see that is different from handle and from non-drivers, the distribution of drivers measures. And here is the consensus of all superpathways. You can see also that is different, especially between this closeness and clustering. It's very different to non-drivers and the handle samples. Here we have the other pathways considered, so chromatin organization, circadian clock, developmental biology, and gene expression. So the comportment, the behavior here is the same. You observe clustering between us and closes that are very different between handle drivers and non-drivers. And we had this first analysis. So what we also did, we considered network resilience. So we are interested to the behavior, to the node removal, in order to maintain the network, the biggest component network. So here, if I remove this node here, the biggest component compound, we have only one, but it's on to illustrate, this removal of this node has no impact, because when I remove this node, I lose only the links concern this node and only this node. But when I remove this node here, for example, you break the network in two, and the biggest component now is quite different from this one. So we also are interested in the comportment of this kind of removal, consider drivers, non-drivers, and you look how the network are the comportment of this dynamic. So here to illustrate, so you have the chromatin organization. So you have non-drivers, hubs, and drivers. The black line represents no impact in the network. So we can observe when we remove non-drivers and hubs, non-drivers and drivers, the network has a small impact. Here, it's the portion of the network we remove, and here we compare the initial biggest component compound, the number of nodes, and the actual biggest component compound. So you have a very small impact when you remove drivers and non-drivers, but when we remove hubs, you have a great impact. So you did this removal a lot of times. So because of that, you have these vertical lines. This is the standard deviation. So we repeat this removal. So you did that to all networks. So we have 3 groups of different comportments. So here you have the 3 chromatin organization, gene expression, and also per pathways. The 3 we have the same. That is the hubs. We have a great impact on the hubs removal, and drivers, no drivers. It's always practically you didn't had impact in the keep the network. It's keep the network connected. Another group was the very resilient networks, pathways, DNA repair, reproduction, and developmental biology. They 3 are very, very resilient under attacks. So here you had 11% of removal, but the network also was about 11% of the connect components. So here's after that you had 84%, 16% of removal. So you can saw that practically the network, the pathway is not break. So reproduction, developmental biology had the same comportment. And the next group, programmed cell death and circadian clock, you had a very interesting comportment when we remove drivers. So here we observe that drivers break the network more than hubs in the beginning, and here after about 9% of removal. So circadian clock also driver genes had also an important comportment here concerning the resilience of the network. So when can we observe with this initial work? That is, we observe that we have significant differences from centrality measures between drivers and non-drivers. You saw that clustering between us and also closeness is very different between drivers and non-drivers in the most of the pathways. But when you saw the network resilience, we observed 3 different kinds of comportments. So you had the driver impact similar to non-driver, we had this remarkable resilience, and also great driver impact in these 2 pathways, in these 2 networks. So what we are planning to do, because we think that drivers may be a topological hole in these pathways. So we will show that maybe they can have these particular holes. You here use the centrality measures, but you are planning to vary, to diversify topological analysis, because when you look at the resilience, you found 3 different comportments. So you plan to expand the topological study to these networks and the impact of drivers in their structure. Other topological analysis, such as persistence homology, can help us to understand these behaviors in paths like DNA repair, that is a very resilient to driver attacks. And so we expect that, in fact, drivers can have a topological behavior. So now you are looking this persistent homology, and you are looking that they play roles in topological structures of great dimensions. So you are in this moment studying this kind of topological behaviors. And if you have successful in this analysis, maybe patterns found can be used to identify and validated unknown drivers. So you are in this planning to do this and investigated the central role of topological central role of the driver genes in this kind of networks. So it's all for today. I'm very happy to be here. I would like to thank again all the organization, especially Stefanella, that's a new colleague and friend. We are now working together. Also, the other participates, or the other organizers too. The audience, thank you to listen to me, and I would like also to thanks my group here. It's also a nice group, and I'm very happy to work with him. Thank you very much.
Thank you so much, Cincia. So let's thank Cincia all together. Wonderful talk as always. I think I recruit you for a mini course for our next semester.
I have to practice my English. My English is not so good, but okay.
No, it was very good. Because it's very nice, incredible material that is very useful for analysis. We know very well in our problem, but I see also in this other cancer problem, in epidemics, and it has so many applications, you know, even to music now. So, so questions, some questions from the public. Let's see. Huh?
Yes, do you listen to me?
Yes.
Okay, great. Congratulations for your talk. I would like to have the opportunity to follow the course you are going to present here. Invitation of Stefanella. So about the super pathways that you define, you define one called gene expression pathway. My question is the following: the other pathway, all of them also has gene expression involved. Then my question is, how independent are these pathways?
So in fact, the most difficult to have there are the biological meanings. So these pathways are defined by hectome. So we took it in this set of pathways from there. So the biological meaning, I know that there is gene expression in all genes, in all tumors, but in this information, the intersection of these, I think I don't know if there is intersection between the 26 superpathways. I think it has, because you have the genes that are in all superpathways. So this biological meaning, we took in the database and we worked with the data you have there. So the biological meanings, because you are a group of mathematicians who don't have biologists. So it's in fact a difficult to have to talk with, to have this validation, the meaning of things, biological things. It's a next step. I think I will do this because it's important to the progression of the work. So in this stage, we only took the data as in reactome. So there is no biological meaning doing for us. So analysis, biological analysis, only network analysis, and biological meanings for us is now another stage.
Here we have Flavia Carneiro, that she's a biologist, and she would like to use these learners. So I think you two should talk.
Yes, it's very good. Good.
You should talk because she's missing your part, and you're missing her part. So I think.
Okay, thank you. Yes.
Actually, I would like to thank you for your talk. Acho que tem que desligar o microfone. Because we are together here, same room. Okay. Thank you for your talk, and I would like to ask you, I don't know if I missed it, have you analyzed cell proliferation pathways like microkinase or something like that?
No, I not listened very well. So cell proliferation, I don't know. We only.
If you analyze the pathways related for cell proliferation.
No, you choose the pathways with more percent of drivers, because we removed some nodes, some drivers, so the pathways with not very quantity of drivers are not interesting to us. So we choose the high percent of drivers. The 6, 7 most with most drivers.
Okay. Yeah, because I was thinking that it could be interesting to analyze the cell proliferation and pathways because of cancer, but you select by the drivers, right?
Yes.
Okay. Thank you. Oi, tá ouvindo? Hello? Hello?
Okay, so thank you very much, Cincia. We have some audio problem here. Thank you very much. Try to connect. This hybrid mode is a little bit complicated. So I guess there is future collaboration here opening up. And we hope that Cincia also can come to Rio in person, so we can all see each other. And now we are going to have a break. No? No. Yes. No. No? No? Okay. So now having a break. Okay. So someone else is going to present Flavia. Is her? Yes. No. Okay. The floor to Fabrizio.
Hello. Good morning. Okay, it's my pleasure to introduce Dr. Carneiro. Dr. Carneiro holds a degree in biological science from the State University of Rio de Janeiro, and a PhD degree in genetics and molecular biology from the State University of Campinas. She has experience in genetics, biochemistry, molecular and cell biology. Dr. Carneiro did her first postdoctoral work at the National Cancer Institute in Rio de Janeiro, and her second postdoctoral stage at Columbia University in New York. She's currently a researcher at Oswaldo Cruz Foundation, in the Center for Technological Development in Health in Rio de Janeiro. In addition, Dr. Carneiro is a visiting researcher at the National Cancer Institute in Brazil, Rio de Janeiro. She works on research projects to produce recombinant proteins in mammalian cells for therapeutic purposes, identifying new therapeutic targets for acute lymphoid leukemia, and study the WNT signaling pathway. So, Dr. Carneiro, thank you so much for accepting our invitation. Please, you can start your talk.
Thank you very much. I will. Eu preciso was disabled participant screen. Oi?
Also disabled participant screen share. Não consigo autorizar. Sou host.
Seconds. Can you see my screen? No? But, gente, eu tenho que olhar o meu PowerPoint aqui, eu não estou conseguindo.
Está no modo vídeo, não?
Pois é, mas está meio estranho aqui. Só meu computador está meio... Porque eu preciso... Tá, mas foi depois que eu conectei isso aqui.
Tira e coloca de novo.
É melhor fazer assim, projetar.
Mas tudo nos áudios.
O áudio, a pessoa vai aqui na nossa defesa.
Porque ele bagunçou meu computador. Deixa eu ver se eu consigo agora ver a minha...
Você está aqui, não precisa.
Eu já estou. Peraí, deixa eu ver lá. Okay, now it's okay. Sorry. Can you hear me? Yeah? Okay. So, first of all, good morning, everyone, and I'd like to thank the Can I continue? Okay. So, good morning, everyone. First of all, I would like to thank the organizing committee for inviting me to give this talk today. And so I am not a bioinformatician, I'm actually a molecular biologist, meaning I am a wet lab researcher. And some of the organizers asked me to talk about some wet lab techniques that we are using in the lab to validate some results that we are getting with bioinformatics analysis. So today I'm going to talk about these techniques and try to show you how much these two words, the wet lab and the dry lab, are highly connected and how much we need each other. So the main points that I'd like to discuss are how are the data that feed bioinformatics generated? And also, how can we validate the results of a bioinformatics analysis? And here I am talking about especially about the transcriptome and the interactome, because we are starting a project now in the lab that is a combination of these two omics. And how can we validate the results? I will focus on techniques that you use to validate if a gene is an oncogene or a tumor suppressor, or if we can target a gene for cancer treatment. So we can look at the integration of the wet lab and the bioinformatics as a virtual cycle where the wet lab generates a lot of results, especially through high throughput biology, and these results are important to feed the databases. And then we have the genome, transcriptome, proteome, interactome, metabolome, methylome, and other omics. So then bioinformatics analysis and system biology analysis generates more results for us. And then we needed to ask ourselves, are those results enough to validate our questions, or do we need to do further validations? And if we need to do further validations, we go back to the wet lab, perform more experiments, and then we can feed again the databases, and then the cycle continues. So, as I said, I will focus on the transcriptome and the interactome. And the transcriptome is the set of our RNA transcripts, including coding and no coding in a cell or a population of cells. So scientists can perform new transcriptomes and analyze them in order to gain a deeper understanding of what constitutes a specific cell type and how that cell type or cell normally functions, how changes in the normal levels of gene activity may reflect or contribute to a disease. So transcriptomes can generate a genome-wide picture of what genes are active from. So first, we need to prepare an Rna-Seq library, and actually there are several protocols for that, and it depends on the platform that's being used for sequencing. So first of all, we isolate the total RNA, and it can either be from tumor sample or normal sample. If you want to analyze a specific gene, it can be from a knockout sample or wild type sample, or if you think that a drug are interfering with the cell transcriptome profile, you can treat the cells and analyze no treated cells versus the treated cells. One thing that you should have in mind is that most of the RNA is going to be ribosomal RNA plus transfer RNA. Only 3% of the RNA will be messenger RNA. And this is the one that most people are interested in because it's the RNA that codifies for proteins. So we have to try to actually enrich your sample for the RNA that you are interested in, either microRNAs or messenger RNAs. Then there are several steps that have to be done, like reverse transcription, adding sequencing adapters. These adapters are related to the platform that's being used, PCR amplification, and then you can actually sequence in your library and analyze the results. So after that, I found the interesting results, and now what? So here we have two situations. I know what I'm looking for, and I don't know what I'm looking for. If I know what I'm looking for, I can ask important biological questions. Is the gene important for tumorigenesis? Is the gene important for metabolism? Is the gene important for inflammation, or any other biological question that you may would like to answer? Or I don't know what I'm looking for, and in this case, you can perform a pathway analysis to detect relevant groups of related genes that are altered in cancer samples in comparison to the controls. Pathway analysis is a very useful tool that helps a lot to analyze data from high-throughput techniques. And also pathway analysis help to generate new hypotheses, design experiments, and further validate new findings. We just have a talk right before talking about pathways and networks. So how can I validate my findings? So to find or verify the protein function, you can either downregulate or upregulate a gene. So if your gene is upregulated, you can downregulate it and analyze the phenotype. You can do that taking advantage of the Rna interference mechanisms, introducing into the cell is more interfering RNA molecules or short hairpin RNA molecules. So here we have an idea, a very simple scheme of how the RNA interference works. The RNAs that trigger the RNAi, they can be either microRNA primary transcripts or double-stranded RNA. The microRNA primary transcripts are first processed in the nucleus by the enzyme DROSHA, giving rise to the pre-microRNA. So double-stranded RNA and pre-microRNA are then cleaved by dicer and small interfering RNA. And these RNAs are then loaded into the RISC, that is the effector complex, and during the RISC assembling, this RNA is unwound. And then we have here a single-stranded RNA that can target the messenger RNA by complementary base pairing. And in this situation, here we have two situations. If the match is perfect, the RNA is cleaved. If the match is not perfect, if there is a mismatch, what happens is that the translation is blocked. mRNA is stable, but does not codify for any protein because the RISC complex is stuck on the target. But in both situations, there is a decrease in the protein levels. You can also use the CRISPR-Cas9 technique. The CRISPR-Cas9 technique is actually a natural occurring mechanism that bacteria use for a defense against virus. And it requires a single guide RNA that is loaded into the Cas9 enzyme, and then the complex Cas9 and single guide RNA can all target the DNA, and it happens through basic complementary base pairing here. And after this hybridization, the DNA is cut. It's a double strand cut in the DNA, and the DNA repair machinery in the cell will try to repair the damage. So it can do that by adding or removing nucleotides, and then it can disrupt an open with frame of the gene, and then you don't have the final product. Or if you want to make a transgene, you can also insert into the cell the sequence that you'd like to be integrated, and during the repair, the cell can integrate the sequence, making then a transgenic DNA. If the DNA, now we can see a situation where the gene is downregulated. So if the gene is downregulated, you can upregulate the gene. And you can do that by cloning the cDNA inside of a plasmid. And here we have protein that I studied in the past, cloned with a GFP gene as well, and then we inserted these plasmids into the cell that will overexpress the protein of interest and also the GFP. We use the GFP here to track the efficiency of the translation. As we can see here, no transfected cells and several transfected cells showing the expression of GFP. So after that, if you downregulate or upregulate your gene, you can ask your biological question, which in my case is the gene important for tumorigenesis? And then we should do a set of techniques to try to prove our hypothesis. And one assay, which is very simple, is a proliferation assay, that you can actually just count the cells in different time points, comparing your control cells and your knockdown cells to see if the knockdown has any interference in the proliferation. Another important assay is the apoptosis assay to measure the rates of programmed cell death. And the rationale behind this, and actually what you do, you stain the cell with an Exim5 coupled to a fluorescence dye, and also with PI, which is propidium iodate. So the rationale behind this assay is that live cells, phosphatidylserine here in red, that is part of the plasma membrane, is facing the inner part of the cell. So we know the apoptotic events, the phosphatidylserine then flipped out of the external side of the membrane and then can be recognized by an Exim5. So in the early apoptotic events, the cells can bind an Exim, but don't are stained for PI. So in late apoptotic events, what happens is that the membrane loses its integrity, and then the PI dye can enter in the cell, and you can measure and monitor the whole process by flow cytometry. And here you have the live cells that are negative for an Exim5 and negative for PI. And here in this square, we have cells in an early apoptotic stage, which they are only positive for an Exim5. And here we have cells that are positive for an Exim5 and PI showing cells in late apoptosis stage or necrosis. Another important assay that we can do is the colony formation assay, and it actually measures the capability of a single cell to grow and form colonies. So here we are analyzing cell survival and also its capability to undergo limited proliferation. So here the authors compared a control and also their cells that are downregulated for the MET gene, and we can see that the downregulation of the MET gene decreases the ability of the cells to produce and form these colonies, suggesting that the downregulation of the MET gene is important for survival and proliferation. So another important assay, other important assays are migration and invasion, and they are crucial because most of the cell deaths related to cancer are because of metastases, and then we can in vitro analyze if a protein is important in this process. So one very easy and cheap test is the wound healing or scratch assay. And in this case, the cells just grow to confluence to form a monolayer. And then we can just use a pipette tip to do a scratch or a wound in the plate. And here we can see these wounds. And then we analyze by the time the migration of the cells to fill this space here. Another important assay for cell migration is the transverse migration assay, and also the transwell invasion assay. So in this case, we have a transwell here, there is a membrane here, and we can see the ability of the cells to migrate into a chemoattractant gradient. And we can also transform the transwell migration assay into an invasion assay by simply adding here on the top extracellular matrix materials. And then you add the cells on the top of this extracellular material, and then you can analyze the invasion. So what you do is you take the membrane that's down here, and we can dye this membrane using crystal violet that stains the cells. And again, comparing here, we can see the wild type cells and the cells that are down-regulated for the MET gene. And you can see that this gene is important for migration, because when it's knocked down, the cells decrease their capability to migrate. And finally, what is very important as well, we can do in vivo assays. And in this case, the authors, they were analyzing the importance of the NA-FAT gene as an oncogene. What they did is that they inject cells overexpressing NA-FAT in these actinic node mice, and then they analyze the formation of tumors. And then we can see here, compared with the cells containing the empty vector, that NA-FAT2 was able to induce the tumor formation, suggests that in this model, NA-FAT is an oncogene. So despite the advances in sequencing genotyping, in genomes and transcriptomes, we still understand a little about the cellular mechanism that mediate phenotypic and tissue or cell type variability. A mechanistic understanding of how gene products interact with each other is crucial to figuring out many cellular functions, and the interactome is helping us with this task. So to get and have the database results, several experiments were done to generate these interactions that then were in this database. So where do the data come from? This data comes from many high throughput or in low throughput techniques, and one of these techniques is the is2 hybrid. is2 hybrid represents the only binary protein-protein interaction assay that can bind, that can be operated at a sufficient throughput to systematically screen the human proteome for binary protein-protein interactions, and it is based on a transcription of factor that is split in two parts, the GBD, which is the DNA binding domain, and the activation domain. And what happens is that a protein is fused to this domain, and another protein is fused to the activation domain. The protein that is fused to the DNA binding domain is called the bait, and the protein that is fused to the activation domain is called spray. And in order to interaction to happen, the two fusion proteins has to be inside the same yeast cell. So to detect the interaction, what happens is that we have reporter genes, but when the prey and the bait interact, they put the DNA binding domain and the activation domain in close proximity, and then we have the activation of the reporter genes. In this case, we used the LAC-Z and the HIS3, and the LAC-Z will confer a blue color to the colonies in the presence of the reagent XDAO, and the HIS3 will lead the cells to grow in the absence of histidine. So here we have an example of a binary protein-protein interaction that we tested using the is2 hybrid system, is a number 2 here. So we can see that the colony is able to grow in the absence of the histidine. We are testing this interaction here in NF2-1 and IRF2-BP2, and the colony turned blue in the presence of XGAL. The same is not true for the negative controls that cannot grow in the absence of the histidine and are not blue in the presence of the XGAL reagents. Another technique that is widely used to detect interaction is affinity purification, followed by mass spectrometry. And in this case, we mobilize our bait in a matrix. We incubate this matrix with a protein extract, and then we washed out, and then collect the protein partners, digest this with a protease, and analyze by mass spectrometry. Another important technique is also co-immunoprecipitation. It's very similar to affinity purification. And in this case, we have our lysates and then our protein of interest here in blue. And we add an antibody that's able to bind this protein. And then we add in the protein extract a bead that is coated with protein A or protein G that has high affinity for the Fc part of the antibodies. And then we pull out the whole complex, wash out the unbounded proteins, and then collect these to analyze. And here we have two situations. We can either, if we know what we're looking for, we can perform a Western blots and identify the proteins with specific antibodies. And in this case, I was confirming the interaction between IRF2BP2 and NAFAT1. So IRF2BP2 was able to pull down NAFAT. So we can see that the interaction was real. And also, if you don't know what you're looking for, you can perform again an analysis by mass spectrometry. So the interactions found with these techniques were important to feed the interaction database. And nowadays, we can easily find a network and an interaction analyzing these databases, but still many questions can remain, like, is this interaction really real in my research model? So we can confirm if the interaction that is in the database is real, is really real. We can do endogenous co-IP in the specific cell type, or adding or not some stimuli or any other questions that you may have. And how does the interaction help me to understand the protein function? And in this case, we can see if this interaction is involved in any pathways that can give us a clue about the function of the protein. So in the next few minutes, I would like to talk to you about the project that we just started in the lab. So as I said, is a combination of the transcriptome and the rectome in order to try to find new targets for TC acute lymphoblastic leukemia. So here we have the instance of leukemia in Brazil, the proportional distribution of the 10 most common types of cancer. Leukemia is the 10th more common cancer in men, and it occupies the 12th position in women. However, leukemia is the most common cancer in children under the age of 15. As we can see here, we have that leukemia is around 30% of the cases in both estimated incidence and estimated mortality, according to the GlobalCan data source. So there are several types of leukemia, and different hematological malignancies originate from different cells. For example, acute myeloid leukemia originates from hematopoietic stem cells or other very premature progenitors. And T-cell acute leukemia originates from thymocytes in the thymus. So the T-cell acute lymphoblastic leukemia is characterized by an immature phenotype of developing T lymphocytes in the thymus. So TO is a heterogeneous disease. So we can see here several subtypes that are characterized by overexpression of transcriptional factors that suffer translocations in the chromosomes. So these are some of the subtypes, and you can also see here genes that are mutated or suffered any other genetic alteration. And what's important to know is that thymocytes undergo different stages of differentiation within the thymus. So we can hear the thymocyte differentiate until it reaches the form of mature lymphocytes. And the subtypes of the TO can be correlated with these different stages of differentiation. So our goal is actually trying to find new targets for this type of leukemia. And what we did so far was we analyzed the Rna-Seq data from databases from cell lines that were originally T-cell acute leukemia, and also Rna-Seq data from normal thymocytes and also mature lymphocytes. We mapped the reads to the coding sequence from proteins from the ABI interactome. And then the tumor and control read count files were normalized according to their corresponding coding sequence. And then what it did was we subtract the normalized Rna-Seq data from the Rna-Seq data from the cell lines to calculate the difference expression. So this subtraction gives this a curve that has this shape, which was similar to a Gaussian curve, and then we determined a threshold above which the genes were considered upregulated, and then we analyzed these upregulated genes according to their number of connections using the intact interactome from EBI. So the idea here is to select genes that are upregulated in these cell lines and also highly connected, meaning that they are hubs. So we compare the controls and the cell lines, the Rna-Seq from the cell lines, we use Jupyter and Multi4, and also all these cells that we use as a control, meaning the thymocytes in different stages of differentiation, and also T lymphocytes in a mature stage. So we did a pairwise comparison among between Jupyter and each of the controls, and also a pairwise comparison between Multi4 and all the controls. So so far, we are actually trying to validate our targets. So what we found was that the potential new targets are similar when we compare the cell lines with the different controls. So we did not find a huge difference between thymocytes when we use thymocytes or mature lymphocytes in the comparison. Some targets are the same for both cell lines and others are specific to each cell line. We identify genes that encode proteins with functions related to cell cycle control, proliferation, signal transduction, autophagy, chaperones, and histone modification. We actually found so far new potential targets for T-cell acute leukemia, and these targets are being validated in vitro in the lab. So I'd like to thank Nicholas Carrolls, that is my collaborator in this project. He actually did this analysis to select the targets, and also my students, Rafael and Patrick, that are doing the validations in vitro, and also members of TDTS, and also the Interdisciplinary Medical Research Laboratory at Fiocruz, and the Program of Immunology and Tumor Biology in INCA, especially Dr. João Viola, and the founding agents. And thank you for the attention.
Thank you very much, Dr. Carneiro, for the great talk. Thank you for accepting our invitation. Thank you so much. I'm going to ask the audience, I think that you have a question. Stefanella, please go ahead.
Try to speak. I have a question. It was very interesting. Sorry about the confusion at the beginning. I didn't have an idea. I couldn't hear. So I would like to know, well, if I can, I would like to know this data about the importance of the leukemia in the young ones. Is overall, is of some country, this as compared with different social classes, or this 30% is universal or is specific of some country? The data are more.
The data are universal. I mean, at least the countries that they have access there, but it's not a specific country. It's a global estimative.
Because just to know if there is some epigenetic effect on this 30%, this was the impact. You know, what started this leukemia? This is my question. You know, if there is something that is epigenetic or not epigenetic in this.
The children.
Yes.
Yeah, in the children, we can have more the genetic effect, but sometimes we cannot identify a specific genetic source, actually. But the thing is, children have less cancers, so in general, but leukemia appears to be an important one in this group of people. But in general, we can try to find a genetic cause, but sometimes we don't have like a hereditary genetic cause. It's a somatic mutations.
So this study that you're doing could eventually in the future, not immediately, help prevention or in a kind of, if not prevention, some therapy to better deal with the progression?
Yeah, the idea is to find these new targets, because what happened is, especially with the acute T-cell leukemia, after 5 years, we have 20% of people that get sick again, and for those people, there is no very good treatment so far, and most of them actually die. So the idea is to try to find new targets using the hub idea. If you can actually identify and do, using other medications for these new targets. So the idea is actually trying to find new treatments. And even for people that are treated now, the treatment for leukemia is very long, it's toxic. And the idea is actually trying to find an alternative for what we have here.
Okay, thank you. Thank you very much.
I have one question actually. On top of Stefanella questions, those databases that you have access, those publicly, the leukemia data is split by age, or that is the data you used are taking into account this part of the percentage that you use it. So, or are you using like adult data, and you can, if you already work of those different data, is there any difference at all? So you really have to take into account the age in the samples of your data to have.
Yeah, it seems so. It seems that the leukemia is different in children and in adults. But what I did so far, I analyzed Rna-Seq data from cell lineage, which was cell lines, not patients. Although I have a list of patients that I have the transcriptome, and I actually separate them by age and everything and subtype, but I didn't have the time to analyze them. So we started with the cell lines. Because first of all, we need to validate if our method is working. So we will find the targets, we will test in the cell lines if the cell stops proliferating, like with all those techniques that I mentioned. And after that, we can analyze the patient's data, but first we need to validate the method. But these cell lines, I think they came from teenagers actually that had leukemia. What I mean, they are cell lines, but still, and yes, I think it's important to look by age as well.
Oh, thank you, Flavio.
I don't know if I can use my computer. Can I talk here?
Yeah,
okay,
I can.
Great.
Yeah.
So I have a comment and a question. First of all, the comment is that your talk is essential for anybody that I think to start to work on this, because especially people that work with modeling, they must to know the origin of the data, how the data are generated, and your talk is a great contribution for that. Thank you very much for that. And my question is in fact a general question, because you said that it has been hard to identify genetic cause for leukemia. And I have been discussed with Dr. Eliana Abdelai. In fact, this is idea that she presented to me my first meeting with her, maybe 5 years ago, Inca. That is the following. We must think more about regulation. The idea is that cancer is more than mutations. Of course, mutations can generate dysregulation in signaling network. However, dysregulation in signaling network can also make the cell to reproduce with more frequency. If the cell is reproduced with more frequency, it can generate mutations.
Mm-hmm.
It means that we can start to think mutation is a cause of cancer, or cancer is a cause of mutations. Of course, if a patient has cancer, mutation will contribute to make the sickness to advance, to make the sickness more serious. But I would like to hear your comments about that. How important is other factors besides mutations?
Yeah, so first, thank you for the question. Mutations cause cancers and cancers cause mutations. I mean, both things actually happens. So the question is about other causes that are not mutations, right? Other cause. I mean, as I know, we have like mutations as a basis of many dysregulations. You can also have epigenetics alterations, right? But people will still think that, these alterations in epigenetics are also caused by mutations in any genes that dysregulate the epigenetic modifications. So we can think about cancer as a genetic disease, as a epigenetic disease, as an immunology disease, and somehow because we have the immune system, having the immune system working well helps to avoid cancer. So I think we can think about cancer using several like ideas. But in my opinion, we still have mutations as very important cause, because if you mutate, we can upregulate other genes that actually are not mutated, but they are just upregulated because another gene was mutated. So I think it's difficult to forget about mutations and think about think about mutations as a separate thing. I don't think it's a separate thing. It's maybe on the basis, but I think we have to think about the immune system as well, and epigenetics as a big picture. I don't know if I answer what you wanted to know.
Great. You say we must think with a system biology mind. Thank you very much.
Okay, let's thank the great talk of Dr. Carneiro. Thank you very much. Thank you for accepting our invitation. And students, thank you all for attending this morning session. We'll be back at 1:30 p.m. with the talk of Dr. Nakaya. Thank you so much.
Thank you.
Alô? Alô?
Sobrou só eu aqui da última palestra para agora.
Você ficou aí? Essa não foi nem almoçar?
Não tô te ouvindo, Helder.
Ih, caralho, peraí. Peraí. Alô, alô. Ué? Alô, alô. Pô, né? Eu tô. O teste aqui tá mostrando que tá funcionando. E o teste diz que o mic funciona. Vou fazer de novo então, vou tentar trocar aqui o microfone.
Ué, que estranho.
Alô, som. E agora? Melhorou? Alô? Melhorou? Alô? Alô, som.
Então o problema sou eu.
Ah, o Dimitri escuta aí. É, não, acho que é você. Dimitri, você me escuta bem então, né? Ah, beleza.
Ah, bem tonta. Eu tinha desativado o som da água porque eu fui almoçar. E aí esqueci de ativar de volta.
Ana.
Tomando a surra da tecnologia.
Pois é. Diz uma coisa, a galera fica se encontrando aqui no Zoom ou ela no YouTube?
Eu não sei direito, Helder. Eu acho que tá um pouco dividido. Porque, assim, no primeiro dia, o que aconteceu? Tinha uma plataforma onde iam transmitir, né?
Aham.
E só que aí acabou não funcionando. E aí eu falei com o professor Marcelo, que ele está participando do comitê de organização, e ele me mandou os links do Zoom. Mas a organização em si, eu não recebi nenhum e-mail deles. O que eu recebi de e-mail deles foi o YouTube. Então pode ser que uma parte esteja no YouTube e uma parte esteja aqui.
E onde é que é o site do YouTube?
Peraí, vou te
Ou
passar.
como os organizadores vão mandar as perguntas para cá? Se tiver alguma.
Eu acredito que sim. É a Stefanella que tá no YouTube da Stefanella. Deixa eu achar aqui, eu já te faço. Eu não sei se chega alguém da organização para eu passar esse anfitrião, mas não chega ninguém.
Será? Tem pessoa que está aqui penduradas.
Hello. Hi, Elder.
E aí, Fabrício? Hi, Fabrício.
Good. Can you hear me?
Yeah.
Great.
Okay, so everything's gonna be in English, right?
Yeah, everything is going to be in English.
There are.
Okay. Okay, everybody. So it's my pleasure to introduce Dr. Hector Nakaya. Dr. Nakaya is a senior researcher at Hospital Israelita Albert Einstein and a member of the scientific committee of the Brazilian Society of Immunology. He is an adjunct professor at Emory University in Atlanta, United States, and was a professor and deputy director of the Faculty of Pharmaceutical Science at the University of São Paulo. He is an affiliate member of the Brazilian Academy of Science. He's a consultant to the Coalition for Epidemic Preparedness Innovations and the International Union of Immunological Societies, and also a member of the Scientific Advisory Board of the European Ebola Vaccine Consortium and the International Institute of Applied System Analysis. So, Dr. Nakaya, thank you so much for being with us today, for accepting our invitation. It's an honor. And please, feel free to start your talk.
Hey, Fabrício, I thank you for this invitation. I think I come almost every year for this event, and I think it's one of the best events for systems biology, and that's why it's such a great opportunity to talk to people interested in this field. So I'm going to talk today about how we use in the lab systems biology to understand the mechanism, a molecular mechanism of infectious disease. And let me share my screen here. Okay. So why we have to use systems biology to understand the molecular mechanism of infectious disease, right? This image here is very cool, right? This was done today by the designer of the lab, trying to show gene network created by artificial intelligence. So this is just a pretty gene network. But the fact is that we have to use systems biology because when we get infected with a pathogen, people may have completely different outcomes. Some people will be completely well or asymptomatic. Some people will have a severe form of the disease, and some people will die of the disease. So, and this happens, these different outcomes, because we have different genders, we have different age, we have different genetic backgrounds, nutrition status, stress status. All these influence how you're going to respond to an infection. And if you're going to see the cells, the metabolites, the proteins, the genes that interact with each other in your immune system, in your microbiome, and if you could measure everything at once, you will see something like this, a hairball, like a huge network complex. And I'm pretty sure you saw this in the first 2 days of the course. What we do in the lab is to try to review which part of this network is important to predict an outcome. So how can we use network analysis tool to reduce the amount of information that we have after you integrate all this information to see which ones can predict an outcome? Well, and where do we take this the data to create these huge and complex networks from? Human studies, from also animal and in vitro studies, normal experiments, but also from the literature. So when they publish these results, we can harvest this information and put in the network. And what I'm not going to show here, it's going to be the project that we use other kind of data, GPS data, Twitter data, to try to identify the mechanism of infectious disease or how the infectious disease spreads in the population. What I had to tell you is that bioinformatics and computational tools does not replace science. This is only like a funnel, something that you use to reduce the amounts of information that you have to read to go and try to fish nice ideas out of this sea of data. So this is what we have been doing the past few years in the lab. We have working completely different projects on long non-coded RNA, systems vaccinology, network medicine, and also in systems immunology of human disease. I'm going to focus only on the ones that are related to infectious disease. We have been publishing paper related most of the diseases that affect the Brazilian population, such as chikungunya, malaria, Zika, dengue, Chagas. And of course, when COVID-19 pandemic started, we shift our efforts to try to understand the disease. I'm going to talk about 2 projects that is published and 1 project that is unpublished. These are the list of the over 30 papers that we have been working with. And the first one that I always like to mention is to show like how fast you can generate a nice hypothesis. And as soon as the COVID-19 started, everyone already knew that there were some comorbidities associated with severe COVID-19, diabetes, chronic obstructive pulmonary disease. So the question was, why different comorbidities will increase your chance of dying of COVID-19? What are the molecular factors that are associated with the disease? Just for you to have an idea of the paper that we published from this, the World Health Organization declares a COVID-19 pandemic on March 11, and on March 27, we submit the paper that was later published in JID. But that's just show that you can use bioinformatics to quickly respond and come up with an idea that is going to be highly cited. It's already over 300 citation of these papers. But this is just to show that we can quickly use data from public repositories. In this case, we use GEO data, transcriptomic data from the lung of people with different comorbidities. And we reanalyze these completely different comorbidities, but in the same tissue, which is lung. to try to see what they have in common, to explain why they increase your chance of dying of severe COVID-19. And we also use a tool called MetaVolcano that we developed in a few years ago. One of the genes that was mostly upregulated in all these comorbidities in the lung was ACE2. And ACE2, you guys know already, that is the main receptor that the virus used to enter the cells. So the hypothesis that we came up with this paper was that people with different comorbidities will have higher levels of ACE2 in the lungs. And when they get infected with the virus, it's going to be more easier for the virus to find on the cells because they are going to have more receptors. And that is going to increase the SARS-CoV-2 replication, which is going to increase the risk of developing severe COVID-19. Very simple message that you can do with public available data. In that time, there was no transcriptomic data of infected people. And then we also ask another question. So are there other potential comorbidities which are associated with severe COVID-19, but nobody look at because people were focusing on lung disease, on disease related to inflammation. So that came up by accident because Leandro, our PhD student, was analyzing data from Barrett's esophagus. And Barrett's esophagus is a condition when you have reflux from your acids of the stomach going to your esophagus, and that changed the mucosa. And that is actually a very common disease, which affects 5% of the US population and 5% of the Brazilian population. And what Leandro did was to reanalyze 8 studies of this disease. And then he found out that the genes that were more consistently upregulated was ACE2 again. So the main receptor of that the virus used to enter the cells. And that could be like a very descriptive and suggestive analysis, speculative analysis. But when we start analyzing the pathways, we saw that the intracellular pH was also affected in the disease. And remember that I told you that the acid from the stomach, because of the reflux, changed the mucosa of the esophagus. So what we are seeing is basically this effect. But the thing is that nobody showed that the acid pH could regulate ACE2. Actually, people knew very little about what regulates ACE2 expression. And then I'm gonna summarize the story. We went to a collaborator, asked them to validate this in vitro. So they took human cells, put in different pH, and when they put it in an acid pH, the expression of ACE2 increases. And if they infect these cells with the virus, they show that the virus replicates more if the pH is more acid, below 7. But what about the clinical implication? Well, we could not pay to recruit people that have Barrett's esophagus, gastritis, and reflux, but we knew that people that have this disease, usually they treat them with proton pump inhibitors, omeprazole, pantoprazole. And then we went to collaborators, which already collected data from these cohorts of people, but they also collected the information about the medicines that they were taking. And then we separate the groups that were taking these medicines because they were treating for this disease. So these are the guys here. And also the people that do not take the drugs. And when we analyze the risk of dying, we see that people that do not take the drug survive more than people that take the drug. And the risk associated with taking the drug, which is having this disease, are higher, even when you adjust for diabetes, hypertension, old age. And then we did this for the North region cohort, but we also validated in a second independent cohort in the Southeast region cohort, showing again that having this disease increases your chance of dying of COVID-19. And that was recently published. But the cool thing here is that we use bioinformatics to come up with a hypothesis that was then validated in vitro. And then we validate in vivo back again in the patients that have or not the disease. So that was a nice example of how we can quickly do this circle and come up with a hypothesis that can be tested. So Covina Transcriptomic Atlas is not published yet. It is led by Jonathan. And why Transcriptomic Atlas? Because there are already over 50 papers on transcriptomics and COVID-19. But why we need an atlas then? Because if you take like 2 random papers and then look at the genes that were described as differentially expressed genes, you're going to see that the overlap between these 2 signatures are very little. So there is a very little consistency of the signatures. So the goal here was to take not only one study, but several studies of the same tissue, and then reanalyze and see which genes are consistently up or down regulated among all these different studies. So for example, if we ask that the gene is upregulating in at least one data set, we're going to see over 5,000 genes. We cannot do any analysis with too many genes. But if we are too stringent and ask to the genes to be DG in 8 studies, we find 0. And then we have to compromise and try to find the genes that are consistently changing. This is more a reliable signature. And we did this for blood. We also did this for liver, kidney, colon, heart, nasopharynx, everything. And now we can connect all these different tissues. What are the genes that are shared by these different tissues? And then this is like an overview of the genes, these dots here, which are changing on severe COVID-19 on these different tissues and what is in common. We also have published papers on systems biology of chikungunya. I'm not going to talk about because this is a published papers. Also on Zika, of congenital Zika syndrome. And what I want to talk is about the yellow fever paper, which is unpublished. This is a collaboration with Aspergillus group. And we collected blood of people during the acute phase of yellow fever during the 2018 outbreak. Some people die of the disease, which are these people here, and some people survive, and we collect blood again to serve as a control. Then when we start analyzing the data, we could see like what are the genes that were highly expressed on specific cell types, which are these ones here, and also are associated with clinical factors such as MELD score, C-reactive proteins, or viral loads. We also, of course, look at what are the genes that change when people die of yellow fever or they survive, to see if we can come up with predictive signatures. And we are now trying to validate some of these findings in experiments. I also want to talk about sexual dimorphism of human disease because this is one of my new passions in my lab. So we know that sex greatly impacts the health and immunity. We know that autoimmune disease is more frequently in women, autism is more frequently in men. And if you look at the blood and measure the expression of 20,000 genes of thousands of men and women with no disease, you're going to see that there are a lot of genes that are associated with sex. Not only the ones that are in the X and Y chromosome, but the autosome chromosomes. And most of the genes are actually related to innate and adaptive immunity. So this shows that this lies at the basis of why there is sexual dimorphism for most of the disease. And that started with Cesar Prada. Cesar was working with arthritis, which is an inflammation of the joints. And it's juvenile because it's giving children under the age of 16, and idiopathic because there is no known cause. And there are over 400 papers that did omics analysis on GIA. And what we knew is that the disease, like most of the autoimmune disease, is more frequently in girls compared to boys. And also that the treatment takes longer to work in girls compared to boys. And what Cesar did was to read every single one of these papers and come up with which are the 90 ones that are important for us to answer this question. What is the difference between boys and girls? And then we start doing the systems biology analysis on these different studies to see what is in common, what is common, what is not. And then he come up with the genes that were consistently upregulated in girls or in boys with the disease. And then he ordered the genes based on if they are more expressing girls or more expressing boys. And there were 44 genes that were highly expressing girls. And if you take the list of 44 genes and then find the pathways that are enriched, we found neutrophil degranulation. So most of the papers stop here. Oh, this is the pathways that are enriched, these are the network, and this is the figure. That's it. And then we discuss a lot about this. But then we ask, like, okay, neutrophil degranulation appears in everything, like all the analysis shows this. So is this really neutrophils? So he went to a single cell RNA-seq data of human blood cells, 45,000 single cells. And then he looked at the 44 genes and saw that the 44 gene was highly expressed on this cluster here, which is in fact neutrophils. Okay, but which neutrophils? There are like different neutrophils. Most of the people look at the mature neutrophils that circulate in the blood, but there are like immature forms that are present in the bone marrow. And in sometimes the inflammatory conditions, these neutrophils go to the blood. So he took a different transcriptomic study where the authors sorted different types of neutrophils. And then when we compare with our signatures, we found that most of the genes were actually coming from bands in neutrophils, the immature neutrophils. Okay, and then this is a trajectory inference, but this is the cool part. You can use these genes as a score and then evaluate, for example, what happens in the blood of males and females when they have no treatment. So females, it seems that have higher score than males. When you treat with placebo for 1 month, nothing happens. But look what happens when you treat with the inhibitor of I1 beta. 1 month, the males goes negative, and 6 months, even more. If you treat girls, nothing changed. So that suggests that immature neutrophils are the reason why girls takes longer to respond to the treatment. Okay, but what is the difference between boys and girls? One of the differences is the sexual hormones that change during puberty, right? And also in the old age. So he went to the public available data of bloods of people, health people on different age groups, and then he saw that there was indeed change over the lifetime of a human being. Which other condition changed with very defined hormones change in the pregnancy? So we took another data set of pregnant women, and then we look at what happens with the genes of immature neutrophils, and we saw that during pregnancy, it starts increasing, and then after birth, it goes back to the normal levels. You see, this clearly shows that sexual hormones influence immature neutrophils, and that can have an impact of the disease, in this case, juvenile idiopathic arthritis. And it's important to mention here that all this, the analysis done here was cost $0 in reagents. Everything was done with public available data, but was not only looking at one data set, but several data sets. Same thing with sepsis. We have been collaborating with different groups that are publishing with sepsis, and then Amanda became interested in looking at what is the potentially difference between men and women with septic shock. So what she did was to collect 9 studies of people with septic shock. She identified who was the girls, or who are the women or the men. And then she started performing the analysis and finding what are the signatures that were consistently changing females and males. And then when she compared the signatures, of course, most of the genes are actually in common to male and female, meaning that these are the genes that are influenced by the septic shock. But there are some genes that are specific to females or males. And when you look at the genes that are specific to females, you see several genes related to bands neutrophils, the immature neutrophils that I just mentioned to you. And among the genes that are downregulated only in females, we saw several genes related to cytotoxic CD8 T cells. We also use the septic data to perform metatranscriptomic analysis. This is Icarus project. And what Icarus did is that when you get Rna-Seq data, you map, the first thing people do is to map the reads into the human genome, and that's going to be the host transcriptome. And with the unmapped reads, they throw away, they put in the garbage. So what Icarus is doing was to get these unmapped reads and trying to align how to detect microbial life. So he does this metatranscriptomics to find bacteria on Rna-Seq data, and he found that several bacteria were associated with sepsis, and several of them were described to be associated with sepsis. He also integrated this with gene expression to show that the genes that were associated with different types of bacteria. And also he found that some bacteria were actually specific to female or male. And there are papers that kind of explain why there is this sexual dimorphism. Spatial transcriptomics, guys, is not directly related to SEPs, but I think this is really cool because we're gonna use this for future papers, future work, including skin of patients with leishmaniasis. But in this first work that we have been doing with Mauro and Leandro, we have data from 3 different sections, this S1, S2, and S3 of the brain of mice. And these are the 3 sessions. And then when we try to use the regions that are described in the book about the mouse region, the mouse brain, and put it to overlap with our data, this is what happens. You see, we're never gonna know like which part of it belongs to this region here, because you cannot overlap what is in the textbook. But you can go to look at the stars, and then try to use reposition, the same tools that people use in to analyze the sky. So we took data, we took a tool, a computational tool from astronomy, and instead of looking at the stars, we're looking at single cells. And then when we do this, we could find a very nice way of dealing with real life data. You see, now we know exactly which region we are talking about. And then we took these 7,000 single cells and start seeing which ones are astrocytes, endothelial cells, neurons, microglia. And this is how these different cell types are distributed in the brain. You see that some cell types seem to be randomly distributed, but some cell types are in a very specific regions. And not only this, if you take, for example, 2 types of cells, neurons and excitatory and inhibitory neurons, and you put everything back again and try to cluster, this is going to be the cluster. So this is red is excitatory neurons and blue inhibitory neurons. And then we can recluster them. We can find subclusters within these neurons. And why we're doing this? Because when we put back these subclusters back into the spatial image, you see that the clusters actually match some of the regions. Cluster 3 are only present here, you see. We do not use the spatial information to cluster these cells, but we use the clusters to put it back here and see that the regions where the neurons are decide of the transcriptome profiling that they have. And of course, you also have, you can look at the gene level. You can look at, for example, 2 genes and see how they are distributed. We also can do more fancy analysis. For example, we can take microglia and then look at the cells that are surrounding each activated microglia to see what are the changes that happens on them. We can also do comparison between left and right parts of the brain. If we take the same cell type but separates left and right and compare, we see that there are genes that are actually more highly expressing on the left or the right parts. And that is kind of striking, right? To see that your brain depends on the sides will express different genes, even if they are the same cell type. And this is just a video showing that you can look at beautiful figures of the genes using the entire image. This is just one gene and the other gene. Well, the lab also is developing several computational tools. I'm not going to talk about them, but I'm going to talk about the knowledge network parts of the lab. And this has to do with the papers that have been published in PubMed, for example. In the year 2000, PubMed has 500,000 papers published in 2000. In 2019, 2020, more than 1 million papers were published every year in PubMed. So doesn't matter the area that you work, you are facing a knowledge tsunami. You cannot read all the papers. You cannot be aware of everything that is being published on these different areas. But although you cannot read 30 million papers, a machine can read them very easily. So we use machine learning, artificial intelligence techniques to read these 30 million papers and come up with knowledge networks that connects genes, drugs, and disease. And then we can use the human intuition and human analysis to try to see which part of this network is important for different disease. So we can use this for repositioning drug to treat for different psychiatric and neurological disorder. This was already published on showing the mechanisms for different diseases using the literature. We also came up with different drugs that could potentially treat Alzheimer. If you look at PubMed, you're never going to see like these antibodies or drugs suggesting treat Alzheimer. But if you go to clinicaltrials.gov or if you go to the internet, you're going to see that there are clinical trials ongoing testing this drug against Alzheimer, showing that there are a lot of things that, of course, the drug pharma companies, they're not going to publish because they want to have the patents, everything. We did this for almost 100 different diseases, and we show that the knowledge change depends on the type of the disease. You can actually see how we study this disease over time. So these are the number of papers published per disease for each year. So this is TB, showing that time most of people were studying TB. Then people were focusing on myocardial infarction, hypertension. And guess what's happened in 1985? AIDS. People start studying AIDS. And then they identify HIV. And then HIV becomes very high. In the year 2000, obesity appears as an important disease. And breast cancer is always on the top until 2020. Guess what happened in 2020? COVID happens. See, everyone became a specialist on COVID. Okay, we also construct different websites that can be helpful for you. For example, if you want to teach how we learn about different topics such as obesity, HIV, there are like thousands and thousands of topics you can search. And then you can change the parameters, increase the speed, but it's going to generate for you how this knowledge was accumulated over time for the topic that you select. So you can play with this if you go to the websites of our lab, or if you can come here, you're going to see. We also have another one that shows how much each country study different topics. Leishmania is one topic that is highly studied in Brazil. And of course, you can also normalize by the total publication of the countries. If you click there, and then you're going to see what happens. Fabrizio, how long I have? How many minutes I still have? Well, I'm going to talk. So Curious is a tool that we are just finishing, and it utilizes machine learning to generate text, but using scientific articles. So we come up with the Alan Turing test, which is a test that they try to evaluate if a human would know if they are interacting with a computer or if a human. What we did was to a version for this for scientists, and I hope you guys can play. We took abstract from Nature and Science papers, and then we separate the titles that were original ones, the ones that were come up with humans. And we also use Curious to read the abstracts and then suggest a title. So these are the titles that are written by artificial intelligence. And we want to know if you can tell if the title is written by an artificial intelligence or a human being. And this is the sites that you guys can go and see if you can find out. So this is one example that if you look at the title, it looked like a human, but actually it was a machine that wrote this title. Here, it looked like a machine, but actually it was a human being that wrote this title. So the tool proved that you can use it to come up with nature and science level titles. Peb, I'm gonna skip this. This is also an unpublished tool that is we are gonna submit soon, but it's too specialized. We have a project on digital epidemiology, which is basically analyzing Google search. And when we use Google search, we can quantify the doubts, the fear, the curiosity of people during the pandemic, for example. And then we can compare this with cases and see if this works in predicting the cases. So for example, if we look at Sao Paulo states, we see that every time people start looking for COVID, the number of cases starts increasing after a few weeks. And then when they stop searching, the cases decline. Increase searching, increase cases. So there is a very beautiful correlation. If you do this for South Carolina, you also see this correlation happening. If you do this for India, you also see the correlation. Actually, if you do this for many countries and many different states, you're going to see that you can almost always correlate the search with the cases, meaning that you can use a very cheap way for surveillance. We are also doing this different approach for Twitter data as well, but I'm not going to talk here. Maybe next year, Fabrice. The last two I want to bring is Tukushi Blast, which was published last week. And that has to do with how the Brazilian unified health system is organized. There are over 42,000 healthcare facilities, primary healthcare facilities in Brazil. And the problem is that there is so many databases that do not communicate to each other. That is, it's bad for science. So we have a database for malaria, one for syphilis. So let's say we have Luis Mendonça that is born in October 6 on 1988, and Luis gets sick and goes to a hospital, and this information goes to the hospital database and sent to the government. Then Luis, a few months later, is vaccinated, and that information also goes to the database. What is important for the scientists and epidemiologists is to try to see if the same person that is in one database is also in another database. That's the record linkage. The problem is in real life scenarios, people do not type correctly the name. So Luis with Z, Luis with S, empty fields, or there is a typo, etc. This is what usually happens, and that prevents record linkage. So our idea was to try to deal with this uncertainty, and which tool is used to work with a lot of data and also data with a lot of unprecision, such as mutation, for example. DNA, genomic tools. So our idea was to convert personal information to sequence of DNAs and then use BLAST to try to make this record linkage. So that's what we did. So that's Tukushi Blast. Tukushi Blast basically converts names, letters, and numbers into DNA using a genetic wheel, you see, which can also be encrypted. And then we use this to compare. If we change the key here, how the wheels happens, M could be GGT or M could be TCA, depends on how you organize the wheel. What we do is then use BLAST, but how BLAST will know like when you're gonna convert this. So let's say let's take a researcher from Fiocruz called Fernando Bossa, and this is how the name look like. If you write Fernando Bossa, these are the changes. You see, if you do BLAST, you're gonna see this match. There is only two mismatch here. If you take Marcelo Bossa, which is his brother, you're gonna see that there is no BLAST results. Now, if you write Fernando Bossa and mother of Fernando Bossa, I don't know her mother's name, and also the Marcelo Bossa, which has the same mother, now BLAST works. But BLAST does not give you the results completely. So if you do machine learning methods, you use the scores, but you also use the coverage to see if you can say that these are linked or not. So we use this to in real life database, we saw which are the most commonly mistaken letters and numbers. We see that, for example, people change M by N because of the keyboard. N is close to M. There are also a lot of changes here. In Brazil, it's very common. In Portuguese, we can do the same analysis for English. We can also use to remove duplicated records. And we simulate the database to see like which tool was the best. And we saw that our tool is 10 times faster with the compared to the best tool. So the best tool takes days, whereas ours take hours. The best tool needs a supercomputer, and we need a laptop to run. And we did this for a real database. And we did this, we found, for example, that there were 84 deaths that were reported in the meningitis, HIV, and tuberculosis database and the central mortality database. This is the traditional records linkage. But when we do our analysis, we found almost 200 deaths, meaning that the government was thinking that 100 people were alive when they were actually dead. We also create a Tukushi translator. You can go here and then type your whatever you want here, and then you convert this into DNA. You can send this to someone, you know, it's just for fun, actually. And to conclude, I would like to say that these different diseases, infectious disease, you can use high throughput techniques, but that's going to generate a lot of data. And computational tools in bioinformatics can only be used to reduce the amount of information, but at the end, you still have to do the hardcore science. You still have to know how to ask the questions. And I'm going to talk about one tool that's called BioFeeds, which we did not publish, which do this funneling. You can use any kind of data, cytokines, proteomics, microRNAs, and what it's going to do is to tell you what are the features that are more important in your biological condition. And it does so by using feature selection methods and also machine learning algorithms to try to select which are the features that are the best. So for example, if you analyze 20,000 genes, you don't need 300 differentially expressed genes to start looking at and see what they do. You can start with these 14 most important genes, and then you start seeing what is the biology behind this. And with that, I'd like to acknowledge my team and of course, acknowledge you all for watching. I hope I did not go over time.
Elder.
Hey, Fabrizio.
So much for the fantastic presentation, for this fantastic amount of work. It's amazing. Thank you so much. Amazing. I'm going to ask the audience here if there are some questions. Well, I do have a question. We know that recently we have seen an exponential amount of single-cell data available in repositories, in public repositories. And sometimes computationally, it's hard to process that amount of data. Can you give us more details on how your group work with that amount of data? That even more if you consider integrative studies, that's a lot of data, and sometimes it's difficult to work with. So how you do that?
Fabrizio, that's a beautiful question that people have been asking this since the microarray area. And that's where I start with omics analysis. And the thing is that the data has been accumulated, and there are some solutions, mathematical solutions to try to integrate them. But I don't know if we can fully trust only on this. For example, single cell, you can also integrate the data, and then you show that in UMAP, it's all clustered together, you know, showing that the integration works perfectly. But we are actually relying the success of integration based on this dimension reduction image. But actually, there are some people showing that this is sometimes not enough because the integration works for some genes, but not for all the genes. So the answer for this is that we acknowledge that there are some limitations on integrating completely different techniques and data sets, and we can also use batch correction methods to try to integrate directly. But in our lab, because of we know that it's hard to sometimes to come up with this integrative solution. In our lab, we decided to work more on meta-analysis. So we do all the analysis with one database, and then we compare the results. So of course, sometimes we can't do this. For example, we generate data from people with leishmaniasis, and we do not have health controls in our data set. We had to include health controls from the skin of other database. And for that, of course, we have no solution. We couldn't do the meta-analysis in a proper way. So the answer is like in our lab, whenever possible, we try to avoid integrating the data in the beginning, because we know that that could bias the results, everything, and work on meta-analysis, because there is a lot of data, as you mentioned, already available. When this is not possible, of course, then we do the integration and then try to come up with the whatever the analysis tell us. But that's kind of our preference, Fabrício. If you have a supercomputer such as your lab, like with the Santos Dumont, then you can integrate everything, you know, like, and then you gain you use the power of the many data sets, which almost become a meta-analysis, because you are putting everything together. But most of the labs cannot do this approach.
Great, fantastic. Thank you so much. I think that Stefanella has a question, so please go ahead.
Yes. How is it?
So Stefanella, we can hear you, but there is some echo because there are two mics in the same room.
I'll come there.
So now, Fabrício, you have to unmute yourself.
Now Stefanella is here.
Yeah.
So
Okay.
can I ask, so great talk.
Thanks.
It's possible to have some reference about that part of research. I mean, everything is interesting. I would like to see that one about the treatment and the men and the women differences, all the study that you did. And this, I kind of forgot because it was so much information. Is this you did also about COVID, or which was the target disease when you did this sensitivity about the treatment and so on?
Yeah, so we for that particular results, we published this on Journal of Leukocyte Biology, focused on juvenile idiopathic arthritis. So this is one inflammatory condition, but we are seeing that the same kinds of signature works with different disease when you separate men and women. So we are trying to see if this signature is also applicable to other disease.
Because we did observe the first year of COVID, when we did some statistic by age group, we did see that somehow men and women were similar, and maybe a little bit more on women in the 20s. But then after, I don't know, 50s or 45 and so on, then the men took over. There were much more cases. And then the question, I mean, maybe naive question, the first question that I thought of is that women go to see the gynecologist and they do take vitamin D, they do take something. And so the first question is that there was also something about this vitamin D helping the immune system somehow, or it was really some gene that was taking over, you know, but why was not taking over in the early age and was only taking over after? So this was the question. So you have an answer?
No, I do not. Actually, that's what I want to tell you. Like, it's kind of tricky to compare men and women, especially, for example, in COVID, where you have also co-founding factors. So for example, men also have more hypertension, more all these diseases that are associated with severe COVID-19, right? Like diabetes, etc. And then women, that also confounds the effect. Women usually lives longer, they take better care of her healthy. And of course, the sexual hormone, the decline of the sexual hormones over age can be one factor that could be associated with these differences in COVID death, but it's hard to pinpoint specifically to genes about that, because otherwise the amount of samples that you have to collect to really answer this question at gene level, it should be like a huge, like, and we don't think we can answer this, but we can definitely see if there are some contributions at gene and cellular level caused by the sexual hormones. And we know that because we collaborate in a paper that was already published showing that the same band neutrophils was one of the differences between men and women with COVID-19 at transcriptome level. But how this is translating to more or less death, it's too big step to propose, you know, like I would not dare to say that that's the reason.
Okay, so great. I would like to talk more in the future.
Yes,
Great.
yes, please.
Thanks.
Okay, any more questions? So let's thank Dr. Nakaya for this fantastic presentation. Thank you so much for accepting our invitation. Thank you.
Thanks.
Thank you very much. Okay, thank you so much. Okay, our next talk, we have 10 minutes. We have 7 minutes. Great. Okay, we will wait for 7 minutes and then we will start the talk of Dr. Tuzinski. Okay, so see you in a moment. Hello, everybody. Okay, so we think that we can go on, right? And it's my great pleasure to introduce Professor Jack Tuszynski. Professor Tuszynski is an Allard Chair and Professor in Experimental Oncology at the University of Alberta's Cross Cancer Institute and a professor in the Department of Physics. Professor Jack Tuszynski received his PhD in condensed matter physics from the University of Calgary in 1983. He is on the editorial board of several international journals and has visiting professorships in Germany, Denmark, France, Belgium, Israel, and China. So, Professor Jack Tuszynski, thank you so much for accepting our invitation to give a talk to us here. And please feel free to start your talk. Thank you so much. Thank you.
Thank you very much, Fabrizio, and thank you, everybody. Thank you for inviting me. It's my great pleasure to be with you electronically. There may be some noises here. I'm in a fairly public space, so unfortunately, this is not perfect, but this is the situation that we all have to put up with these days. So I would like to share my screen. Okay, here it is. And this will be a bit different, I think, because I will be talking about systems, aspects of systems biology, but whereas traditional systems biology is about protein-protein interactions, networks, gene-gene interactions, and all kinds of massive amounts of data, I will be talking about very little known aspects of cellular interactions, and that has to do with electromagnetic and electrodynamic interactions, which I think is very important and very much neglected. So hopefully this will be entertaining and interesting to some of you, but as I say, it's quite different. So the potential applications of these kinds of investigations are just emerging, and I think, as Fabrizio mentioned, I'm involved in cancer research, and that's one of the aspects which I'll talk to you about later on. And there's a plethora of organizations which supported this work over the years, including Novocure, which is an experimental electric technology-based therapy for especially brain cancer. And we've been doing some investigations to find out what the mode of action is, because that's interesting that in many cases the cure seems to be working, and that's not pharmacological, it's electromagnetic or electrical, but it's not very well known what causes these effects. So I will try to explain how I see it, and obviously this is work in progress, very much so, and this is the outline of my talk. I will focus on one particular aspect of cellular structure, but very important in my opinion, tubulin, the protein called tubulin and microtubules, which are composed of tubulin, and we'll talk about electrostatic properties of tubulin and microtubules, their mobility, response to electric fields, also sensitivity to the environmental conditions, which is very important and quite often, again, ignored. We seem to look at the cell as a black box with lots of little elements interacting, but the environment is enormously important. The cells respond to it and change their behavior and function. I will mention later on some experiments. So this talk is a combination of experimental and computational work in collaboration with the people I showed you earlier. So the experiments we did involved also measurements of ionic conduction and also capacitance. So it's impedance in the lingo electricity and magnetism, real and imaginary impedance components. And then there will be a few slides on our experiments in collaboration with other groups on effects of electromagnetic fields in the various ranges from terahertz to near visible and visible. And then time permitting, I will skip to some more recent work, which we're just finishing, a project on quantum effects, which brings some novelty to the field on a different level in terms of tryptophan fluorescence as well as delayed luminescence. And I will try to finish with some forward-looking statements about how cancer cells may differ from normal cells in terms of their electromagnetic dynamics, as well as what I would like to see in the future along the lines of systems biology, but based on cellular electrodynamics as a system in its own right. So this is a bit like comparing, building a structure from brick and mortar and adding to it electrical wiring. So I'm looking electrical wiring, lights, switches and outlets, as opposed to just the stability of the structure. So let me first introduce you to the concepts related to microtubules. Their role in cell biology is very important. They define the cell shape and rigidity. They perform various functions in cell motility, together with another important protein, actin and actin filaments. And crucially, microtubules orchestrate and perform mechanical functions in dividing cells, eukaryotic cells. On all eukaryotic cells have microtubules. You can see here in the middle. And we'll see movies by the end of this talk showing you differences in cell division for cancer cells exposed to electric fields and non-exposed. And finally, microtubules participate in intracellular transport, carrying materials from point A to point B, and providing pathways. The actual process of mechanical movement is performed by motor proteins such as kinesin, and shown here, and that's important too, because electric fields may disrupt this traffic, which is very important to the cell survival. And finally, bioenergetics of the cell is important because this structure here, kinesin and other motor proteins, dyneins, which move on microtubules, also carry mitochondria. So cell energetics is involved very much directly with microtubules and motor proteins. So this is microtubule is a cylindrical protein filament shown here in the center, which I will not go into great detail because that's not the main focus of the talk, but just to give you an idea, they assemble and disassemble asymmetrically. They assemble in a more or less linear process, they disassemble by catastrophes. As you can see at the bottom, there is a computer simulation of the disassembling microtubule where the individual filaments, they're called protofilaments, peel off as if it was, they're called ram's horn. So it's somewhat similar to wilted flowers in a vase, for example. Now, as I mentioned earlier, my interest here in this talk is to uncover their electrical and electromagnetic properties, and therefore how the cells may be affected by that. And so there's a fairly long story behind this, where we, my group and other groups as well, studied resistance and capacitance and signal transmission along microtubules, as well as response to light, visible and near visible. So, but before I start and show you some of these experimental data, I want to point out that this is not an average protein. Tubulin, a dimer shown here on the left, has a very large electrical charge, negative 50, roughly speaking, for a single dimer, so two monomers, alpha and beta, where about 40% of the charge is accumulated on the C termini, which protrude from the globular structure of the protein, and they are very flexible. So that's one of the things I'll mention to you later, that these flexible charges respond dynamically to electric and electromagnetic fields. And the second thing is, so the net charge pointed, shown in the red arrow on the left, about -50 or so. That depends on the specific type of tubulin. I don't want to get into too much detail, but roughly speaking, that is an outlier. The histogram shows you different proteins, all the proteins, thousands or 30,000 or so proteins assembled here in the histogram on the left, and on the right you have the same proteins assembled to show you the dipole moment, because when you introduce a charged protein into a solution, ionic solution, then this protein will be neutralized by counter ions, but what will remain is a dipole moment of the charge distribution. And again, the red arrow on the right shows you the dipole moment of the tubulin dimer, which is somewhere in the 25 to 3500 dBi range. For non-physicists, 1 dBi is, roughly speaking, I don't remember exactly, 1.8 dBi I think is the dipole moment of a water molecule. So this is 3 orders of magnitude greater than that of a water molecule. And water, of course, is surrounding the proteins in the cytoplasm, and that's another story for another day. What happens to water, and we'll focus on just the protein and the ions. So this is just a short summary of why this is important, because of the participation in various cellular functions, cell division, intracellular transport. Also, microtubules are known to modulate ion channels, so the interactions with the external environment are affected, cell motility, signal propagation. And we are challenging here the central dogma of cell biology, that only local interactions play a role, and I think this is going to be quite obvious, because long-range interactions also play a major role in cell dynamics, and long-range meaning hundreds of nanometers to a micrometer or more would span the length of a cell or so, or parts of the cell, which could facilitate signal transmission and reactions to stimuli, as well as affect cell-cell communication. So the ramifications of this kind of thing are quite enormous. Here is an old paper, now more than 20 years old. At the time, it was a breakthrough by Baker and colleagues published in PNAS. There was a computer simulation of the entire microtubule, consisting of something like 1 million or a couple million atoms, and showing you that a microtubule has a very large negative charge. Red on the left in panel A is the entire microtubule, and red is negative charge, blue is positive. So there are some patches of positive charge which may be very important for various functions. And B and C show the front and the back of a microtubule. There's the plus end and the minus end. Plus end is the more dynamic, faster growing. The negative minus end is more stationary. So there's asymmetry, there's an electric field along the microtubule, and there's a huge amount of charge, which will, of course, interact with electric and electromagnetic fields. And that's the main message I want you to take home from these few slides so far. And then of course, what's polarizability? Polarizability is the change in the dipole moment or polarization with respect to the electric field change, the delta P, delta E ratio. And polarizability in the case of microtubules is composed of various contributions, including the stretching of dipole moments, the rotation of dipole moments, the reorganization of C-termini, these flexible protrusions, which are very negatively charged, but they attract counter ions, positive ions, so that whole thing moves with the field. And then, of course, the water molecules, which are lining the surface, and they are not randomly oriented because of the dipole moment. They form organized structures. So all of this we've investigated both computational and experimental. These are some experimental data on polarizability alpha of tubulin, and epsilon is the dielectric constant in blue, the left panel. Alpha is in black on the left panel. And what I want you to see and take home the message is that this is strongly pH dependent. If you studied the literature on cancer cells, for example, you will know that cancer cells produce acidic environment, but they are more basic inside the cell. So there is a difference in pH. And here, even the range of pH from 6.6 to 7.4 affects polarizability, that is response to electric fields, by a large factor, from 9 down to 4 for alpha, and for the electric, the dielectric constant epsilon, the change is from about 4 to 2, and that affects various electrical responses. And on the right, you have also a plot of the change in the dipole moment as a function of pH, as well as the change in the net charge, because amino acids get protonated and deprotonated depending on the pH. At the bottom, you have the reference to this paper, which was published 3 years ago. Earlier work by other authors indicated that microtubules respond actually quite dramatically to electric fields, and these are weak electric fields, 50 volts per centimeter. They are on the order of cytoplasmic electric fields, and they can glide and move along the field line. So microtubules being negatively charged, they will move towards the positive electrode naturally, and they will rotate, reorient themselves. And of course, we can also introduce higher electric fields and then add to the electric field the frequency. So this becomes AC, alternating current electric fields. And the dramatic effects were observed going back 15 or so years ago, with realignment of microtubules shown here in white in these micrograph reorientation and more complex behaviors depending on the frequency, which is interesting. And 3 different effects were identified physically: dielectrophoresis, so that means movements, forces due to the electric field gradients, electroosmotic flows, and thermal conduction. There was some heating effect, so there will be also that. And you can see here in this panel different organizational structures of microtubules shown in white, and the black are the electrodes that send these electric fields in the gap, the space between, and somewhat around as well. So now we have two things to consider. The first is the strength of the electric field, and the second is the frequency of the electric field. And here I've kind of summarized our best guesses as to which frequencies interact with what, and I've listed different mechanisms. For single microtubules, we know a little bit more than organized structures of microtubules. I haven't mentioned that yet, but it's important the microtubules organize themselves in dividing cells into asters, radiating from the centrosomes. You will see that later. In the neurons, for example, they form parallel bundles, and that resembles parallel computing architecture. Perhaps they do perform some computational tasks. That has been speculated. And then they also form the so-called centrioles. You can see at the bottom left corner, which are 9 triplets of microtubules, barrels of microtubules perpendicular to each other. That's also been called the central enigma of cell biology, because the centriole seems to be the organelle that is the first one to divide when the cell goes into mitosis. So where's the signal coming from? What's happening? Once again, microtubules at the center of this stage. And I've listed here frequencies, which we'll talk about in a few minutes, up to hundreds or even thousands of terahertz. Terahertz is quite common these days. You know, in airport scanners, they use terahertz frequencies to find out if somebody's carrying weapons or whatnot. But these are not naturally occurring fields in biology, as far as we know, but they are absorbed by water very strongly. So we wrote a paper a few years ago about postulating various potential conductive properties of microtubules. So these would be our biological wires that may carry signals, electrical signals, maybe even rapidly. Signals could trigger transformations, you know, activity of ion channels, and so on. And we propose that could be ionic. So there's lots of potassium, sodium ions, of course, calcium. Calcium is known to be involved in signaling, and of course, protonic and possible electronic. So far, as far as I know, nobody has demonstrated conductivity in the form of electron flows like in metals, but that's vaguely possible. It's been speculated for decades that that's happening at the cell level. So now the second. So we talked about the strength of the electric field, the frequency of the electric field. Now we'll talk about the strength of the, ionic solution. So that's very important to note, because different cells have different ionic concentrations, and in fact, Mike Levin from Tufts University is a very strong proponent of the importance of ionic concentrations and even transformation of cellular phenotype. So we embarked on a study of the effects of ionic concentrations, especially potassium, on the conductivity of microtubules. And we discovered that there are many combined effects of different types. So microtubules may form a physical barrier that impedes ionic conduction. They may also attract and trap ions. The ions can condense on the surface. They may also accelerate ions in their flows. And finally, the C-termini may cause dynamic ionic fluxes. So a lot of things to uncover, and we summarized the literature on this topic, and going back actually to the 1970s. And the important thing is the last column. Different experimentalists disagree hugely in terms of the conductivity, which is measured as Siemens per meter. You can see here the values ranging over 7 orders of magnitude. But now I think we understand why, because the experiments may have used different concentrations of tubulin, different concentrations of ions, and different types of probes. So this is the paper that kind of got me into this field in 2006 with my colleague Horacio Cantiello from, at that time, Harvard, now in Buenos Aires, your neighbor to the south, I guess. So what this shows you is that, so Horacio was very skilled. You can see in the top right corner 2 micropipettes holding a single microtubule, which is about 20 micron long, and then one electrode is sending a current and the other is receiving it. And you can see here the blue is the input voltage signal, and the red is the electrical signal received at the other end of a microtubule, around 20 microns away or so. And you can see the amplification of the signal as it propagates along the microtubule, which we termed transistor-like behavior. So microtubules in this particular case of injected voltage signals performed amplification like a transistor. So that was very interesting. And we built a electrical transmission line model for microtubules where we envisaged it as a combination of resistive elements because it's a flow of ions, so it's opposed by viscosity of the fluid, as well as the capacitive aspect because you have negative charges on the surface and counter ions forming a double layer. So it's a nonlinear capacitor because it's saturable. And then finally, there is a inductive element because some flows may be actually following the natural geometry of microtubules in a solenoidal fashion. And we estimated these values for a single dimer, and then you can extrapolate to a microtubule. And then almost a decade and a half later, we managed to measure these things with my colleagues. So this is, for some of you who are interested in nonlinear differential equations, the resultant equations are quite horrific when you go into the continuum model, but we're able to solve them by successive integrations and obtained Fisher type solutions, soliton-like solutions, which would kind of explain this this nonlinear behavior. This is the simplified equation for the traveling wave solutions. Okay, and maybe I'll skip that because there were several papers in the series where we made additional assumptions and more accurate representation of the whole microtubule, including a three-dimensional structure and the effect of C termini. This is what I wanted you to now to take a look at, because it's the medium in which these microtubules exist, namely the cytoplasm. And first of all, you have the transmembrane potential on the left, which is on the order of from 10 to 90 or 100 millivolts. But the membrane is about 5 nanometers thick, which means the electric field across the membrane is huge. It's about 10 million volts per meter. So there is a lot of electricity there. And then, as you can see here, this is from Mike Levin's review paper. It shows you how different cells have different transmembrane potentials. So you can almost create some sort of logic around it. And what's important is that cancer cells have a very low transmembrane potential. I mentioned to you one electrical aspect of cancer, which is the acidic, more acidic environment. The extracellular matrix becomes acidic. The intracellular material becomes basic. And here's the second aspect of it, which is the transmembrane potential of cancer cells gets diminished and almost abolished. The more malignant the cells are, the less of a potential barrier, electrically speaking, the membrane presents. And here you have the typical, in the table on the right, typical concentrations of potassium and sodium, chlorine, calcium, magnesium. And obviously inside the cell, potassium is the main ion to take a look at. These are micrographs of the microtubules in our experiments, which were performed outside the cell, so in a Petri dish, constructed and stabilized pharmacologically, and then we measured their conductivity at different concentrations. You can see the meshwork. That alone is this sort of system that one could call a systems biology object, but this is physical. This is not some arbitrary interaction. These are real microtubules in white, and they form very dense structures in at least in these laboratory conditions. And we performed conductivity measurements using a semiconducting characterization device called Keithley with 4 probe and later on 2 probe, we'll see. And we observe these interesting results. The first one is conductance as a function of frequency. We arranged the frequency from 1 kilohertz to 1 megahertz, and we obtained a broad peak at about 110 kilohertz. And microtubules were more conductive than tubulin, which in solution, which were more conductive than the buffer solution itself. Why is this 110 kilohertz important? This actually corresponds to the frequency which was empirically determined to act most significantly on cancer cells in these Novocure experiments, which were performed in between 2004 and 2006. And then now it's a big company in Israel that benefits to patients with glioblastoma, giving them about a year of life more with no side effects to speak of. So maybe it's coincidental, we don't know. Definitely this frequency of electric field has a special effect on microtubules. We published this several years ago. And we continue these experiments with increasing ionic concentrations, bringing them more to physiological levels, and then also increasing the amount of tubulin in these samples, as you can see them here, and making 2 probe measurements. And that's the paper in Small 2020, which I'm going to give you the results of. So the first observation is this, that as a function of frequency, this is capacitance, so imaginary impedance. Microtubules are much more capacitive, so they have a higher capacitance, act as capacitors, really cylindrical capacitors, if you remember high school physics or maybe first year physics, than tubulin, and more so than the buffer solution. And the typical curve for capacitance, for impedance, imaginary part of impedance is actually 1 over omega, and then the coefficient is the capacitance, which you can read off the graphs, as you can see here. On the other hand, the real part of impedance, namely resistance, shows the opposite effect now. Microtubules are more resistive than tubulin, and tubulin is more resistive than solution at these frequency ranges, and also at this high concentration of ions. So something really strange happened. If you paid attention, the story kind of flipped on its head. At low ionic concentrations, microtubules are highly conductive. At high ionic concentrations are highly capacitive. And so what gives? Well, what's the reason for this? And that's basically the summary. Microtubules at low ionic concentrations increase conductivity. At high ionic concentrations, decrease conductivity. So they can play dual roles of capacitors or resistors in a cell. And that may be quite interesting, different from your semiconductor circuitry. This is an element of a biological circuit which can be both as capacitor and resistor. And so we've done, I'm going to skip quickly because we've done the analysis of these results and obtained these values for the single 20 micrometer microtubule. The resistance R is about 2 giga ohms, capacitance 2 picofarads, inductance 7 femtohenries. And that actually compares to reasonably well to computational predictions, which we had done earlier. You can see the table that compares these values, experiment theory in the same order of magnitude. And there is also a scaling relationship. It all depends on the length of microtubules. Resistance increases with length, capacitance too, inductance goes oppositely. All of it depends on concentration. And back to my question, what's happening as a function of concentration of ions? So maybe I'll just I'll come back to this in a minute. I have the slides in a slightly different order, but maybe I'll just amuse you with a few videos in the meantime, showing you that there is electrophoretic mobility. You can see tubulin and microtubules moving to the opposite electrodes, and we measured this effect and found something quite interesting. The mobility as a function of pH changes sign, which means that at around pH 5, which may happen. In the gastrointestinal system, pH goes down even to 2, microtubules become positively charged. I kept talking to you in the beginning, at least, about how negatively charged they are, but that charge is tunable. You can have microtubules which are also positively charged. It all depends on the pH of the solution. So the cell has a number of controls that it can use. It can use different pH. It can use different ionic concentrations. Temperature maybe not so much, but in inflammatory states you have a slightly increased temperature as well. And that also corresponds to the so-called zeta potential, which is the potential on the surface, which changes sign as a function of pH. And so to come back to the question of what happens, we actually did a computer simulation using the so-called Poisson-Boltzmann model equation, intending to elucidate the role of ions in the conductivity and electrostatic properties around microtubules. This is the paper which was published actually a year ago in Frontiers in Molecular Biosciences. And we used these equations. So it was a modified Poisson-Boltzmann equation, which allowed for variable permittivity, ion size. You can look up the paper. I will just show you the results. And the results tell the story. There really is an explanation of what's going on. Basically, at low ionic concentrations, and this is three different parts of the microtubule. Lumen is the interior. Remember, microtubules are cylinders with hollow interior. We call it lumen. And this lumen has also a charge concentration around it. And in this case, the ionic concentration doesn't change that distribution of the charge much in the lumen. But on the outer surface, there is a difference in this range over which ions feel, so to speak, the surface of a microtubule, and even more so for C termini. This is the summary of these results, where you have the on the left axis, you have the bulk concentration of KCl, and so you go from very low concentration to very high as you go towards the origin, and then the vertical axis is the local concentration. That means around the various aspects of microtubules. It changes quite a bit as you come close to the surface. The same for C termini, and the same for the outer surface of microtubules. Okay, so we notice a huge dependence on the ionic concentration, how the distribution takes place. It's either very tight. For high concentrations, the ions are tightly bound to the surface, all surfaces. And for low concentrations, they spread out tremendously. And I think the next slide will show you how this affects. Yeah. So you see, especially the outer surface of microtubules, the middle panel on the left, it shows you that the radial distance of ionic concentration drops down almost to zero. I would say 1 nanometer at about 100 millimolar concentration, and extends to almost 100 nanometers, 100 times greater when you lower the concentration to micromolar, 10 micromolar or so. And now the final sort of message from that paper is here on the on the right. The figure on the right shows you the conductivity of the solution in red, which linearly increases with the concentration of ions, which is called the Kohlrausch law in electrophysiology. And then you have the blue and the green. Let's focus on the green, that's more important. The conductivity of microtubules as a function of ionic concentration. And you can now see why this seemingly contradictory message from our experiments, because at low concentrations, microtubules can be up to 2 or 3 orders of magnitude more conductive than the solution itself, the cytoplasm. Whereas at 100 millimolar, this is a crossover point. At this point, both microtubules and the cytoplasm have the same conductivity. Above 100, the opposite holds true, namely solution becomes more conductive than the microtubules. So if the cell wants to be close to 100 millimolar, it can switch between signals being propagated through the solution or through the microtubules as it pleases if it changes the so it's kind of a phase transition in conductivity. And let me maybe I'll skip this in the interest of time. We were able to demonstrate this experimentally with a compound called P4VB, which is a reporter molecule for pH, and it shows you here microtubules. It basically emits light, absorbs and emits light at a different wavelength and a different intensity depending on the local pH. And we showed here in these diagrams and published last year or early this year that microtubules locally can have a pH by different a lower pH by a factor of 10 or so compared to the solution. So pH in this case is proton concentrations, positive species, ionic species. So it applies also to the ions. Therefore, you can think about microtubules as attractors to counter ions. Let me switch gears now. So far, it's been all about conductivity. In the second half of this talk, maybe less than half, I will speak about electromagnetic effects and the possibility. So this is first terahertz. Terahertz is from about 1 hertz to about 100 or 200 terahertz. That's the range of electromagnetic waves which cause high frequency water oscillations. But not only that, they also interact with proteins, protein dynamics, and with my colleague Frank Hegman at the Department of Physics and his student Cam Howe, we did some experimental microtubules, but they did also very interesting experiments on animal tissue and the response of tissue to electromagnetic radiation in the terahertz region. And this was analyzed using microarrays, and that shows you this volcano diagram where a number of proteins were protein pathways, signaling pathways, and metabolic pathways were some upregulated, some downregulated, and it was actually intensity dependent. So the energy of the pulse, this is pulse terahertz, changed from 0.03 microjoules to 2.4, so 3 orders of magnitude, no, 2 orders of magnitude, and as a result of that, you have at high intensity, you have much more robust response of the tissue. This shows you the affected cancer-related genes, RAP1 signaling, RAS signaling, calcium signaling, cGMP, PKG signaling, and also actin cytoskeleton was affected. And pro-mitotic aspects were suppressed, whereas inflammatory pathways such as cytokine receptor interactions were upregulated, and some cancer-specific genes and pathways were either suppressed, like glioma, and some were activated. So very interesting. This is, I would say, opens an avenue for major investigations. This is a table of the logarithmic response of the different specific pathways. Some of them maybe you're familiar with, I'm sure, things like KRAS and MAPK3 and so on. So all of it, the cell, cancer cells in this case, respond by upregulation or downregulation of some of these pathways. If we knew which frequencies to use and which intensities to suppress some overexpressed pathways, I think that could be a very good therapeutic approach with little, if any, side effects ISA pulses. So there's actually this is non-ionizing and non-thermal. Now, the same technology, terahertz radiation, was used in laboratory conditions to interact with microtubules. As you can see here on the left, you have terahertz off, and the white patches are clumps of microtubules in the sample. On the right, you have terahertz on, and you can see over time, the time axis is on from 0 minutes to 30 minutes at the bottom. Microtubules disappear, and some micrographs here on the right show you that they are broken into pieces. And actually, I have a video here showing you time-lapse fluorescently labeled microtubules. They disassemble, melt away, basically, as a result of exposure to this. So, microtubules being critical to cell division, if we could utilize this kind of technology to basically prevent cancer cells from dividing, that's kind of bingo, we've done it. The problem, of course, is the fact that terahertz is attenuated through the human tissue. So only superficial cancers could be treated this way, melanoma perhaps. And here we show you surprisingly lower frequencies, 0.5 terahertz, are more effective in disassembling microtubules over time. So the time varies from 0 to 40 minutes. Over 15 to 20 minutes, you can basically disassemble all the microtubules in the sample. So maybe I'll just, for the sake of time, I will skip this. We've tried to explain some of it by doing computer simulations, molecular dynamics with my colleagues in Rome and in Prague. And we determined that there is actually a critical field intensity, namely 50 megavolts per meter, which is quite strong, in order to have lasting effects on the microtubule conformational changes, tubulin. So very briefly, without getting too much into detail, we believe that microtubules are destabilized as a result of conformational change taking place in tubulin dimer, the building block that is exposed to this electromagnetic effect. And as it changes its conformation, it basically weakens the bonds between dimers, and eventually the microtubule disassembles. So that's basically our explanation. Now, in the last few minutes, I'll just talk about some more recent effects. We've been fortunate enough to get funding from the Templeton Foundation to investigate the possibility of quantum interactions. So now we're talking about even a higher frequency, higher energy, ultraviolet and visible light. And excitations involve specific residues in tubulin dimers, tryptophans, which absorb in the basically 200. Sorry, this is. I will come back to this because it's okay. This is out of order. Tryptophans. Yeah, here it is. Tryptophans get excited in the about 250 to 300 nanometer wavelength, which is ultraviolet. And there are some other tyrosine and phenylalanine residues which get excited in a similar range, but at a much lower ratio. So most of it is absorbance by tryptophans. And this is the setup that was used to measure the time-correlated single-photon counting spectroscopy in the lab of Greg Scholes at Princeton in chemistry. And these are the results for microtubules and tubulin, which are very interesting, unpublished data, by the way, so you're the first audience to see it. What happens is that the ultraviolet radiation gets trapped in microtubules and tubulin and re-emitted. And here is the very intriguing aspect. The re-emission takes place over tens of nanoseconds, as you can see here. so it's clearly a quantum absorption because it's a laser light at a specific wavelength. And some tryptophan groups are excited, and then we have reason to believe, and I have no time to explain in great detail why, that there are collective excitations. So these tryptophans form a quantum states which spread over several dimers of up to 8, maybe even 10 dimers of tubulin, which corresponds to something like 70 nanometer distance. So I don't know if anybody talked about quantum biology here, but that is much longer span than what's typically invoked in photosynthetic systems in quantum biology. Here we have a quantum system in the form of many tubulin dimers in the microtubule, trapping quantum states over on the order of, let's say, 10 nanoseconds, and the range of quantum state is 60 to 70 nanometers. So that's the result. We're just about to submit the paper to PNAS with these results, because that kind of opens a completely new understanding of quantum biology in the context of not chlorophyll, but also microtubules in eukaryotic cells and so on. So let me just conclude with, I have way too many slides, but come back to mitosis and why we are so interested in applications to cancer. So this is a very intricate way of harnessing microtubules to pull apart kinetochores and through kinetochores chromosomes and divide the cell eventually. This is a video of a dividing cell and mitochondria labeled. So, okay, okay, here is the summary of these things that change between normal and cancer cells. I mentioned to you the transmembrane potential, pH, internal, external, and there's also a potassium concentration change as a function of the transmembrane potential, and therefore, by implication, ionic conductivity along microtubules. So the whole thing is electrodynamically destabilized when you compare normal and cancer cells. How is it, including the pH gradient, which is abolished basically in cancer cells compared to normal cells? This is the paper that changed the whole field, PNAS 2007 by Kirsten et al., and showed that these alternating 100, 200 kilohertz electric fields target microtubules and prevent or delay mitosis. So here's I'll show you two videos of dividing cells in the presence of these fields and in the absence. First, in the absence, here's on the left, just very beautiful, symmetrical, rapid separation of two cancer cells. This is HeLa cells, by the way, and everybody has seen this kind of thing, but it's still very impressive and beautiful. And now let's take a look at the same thing happening in the presence of these TT fields, tumor treating fields on the right. See, yeah, there's this bridge, and the green is microtubules. Microtubules cannot stop connecting the two cells. The cells cannot complete cytokinesis, and well, they eventually do, but our statistics show that it takes them, between 2 and 5 times longer, and some cells never divide, maybe a third, and they go apoptotic eventually. Probably has to do with the direction of the field, and so that's kind of, now I'm going to stop in a minute, just to want to show you what I'm proposing, that the systems biology of electromagnetism in cells should do, and that is basically represent the cell as a circuit. In this circuit, we will have the membrane, the ion channels as separate electrical entities or elements. Then we have the solution as a resistor capacitor. We have microtubules, the cytoplasm, and actin filaments, which I only barely mentioned. And if we could understand the values and simulate the entire cell, maybe that would be the beginning of a different approach to cancer and other diseases, which I'm sure have a lot of changes, as I tried to emphasize, in terms of their electrical and electromagnetic properties. So that's kind of my modest proposal for another branch of systems biology, biology of electromagnetic cells. Okay, so that's the summary. So very different behaviors of microtubules under different ionic conditions, very robust responses to electric fields, electromagnetic fields, and all depends on the concentrations of ions and pH, and possibly also transmembrane potentials, which we haven't really explored fully yet. So I'm sorry I've taken so much of your time, but hopefully there was something of interest to some of you at least. And thank you very much for your attention.
Thank you, Jack, for the fantastic talk. Very interesting. Very interesting. Thank you so much. I will open for questions. The audience has any question? Well, I do have. About therapeutic interventions in cancer. How do you foresee the use of these, let's say, the use of the electromagnetic fields on microtubes on specific types of cancer, those types of cancer that are most difficult to treat? How can we think about that?
Yeah, so you see, maybe I'll step back a little bit. You probably know that microtubules were some of the first targets in cancer chemotherapy, right? Maybe about a third of all drugs used in cancer chemotherapy target microtubules or tubulin. So this is a very good target. However, all these drugs are very toxic, and they cause a lot of damage to normal tissue, various organs, liver, the kidneys, and so on get damaged. So my answer to your question is that if we, basically everything you do to mess up with microtubules in cancer cells is going to help stop or delay cancer progression. And I think what we need to find out is resonant frequencies. And I think this Novocure company in Israel found one such frequency, about 100 kilohertz, and uses it with some challenges, of course, delivering this through the tissue and so on, but it is successful. So my answer to your question is that what we need to do is a systematic scan of the frequency ranges, let's say from low frequencies to even in the hundreds of hertz to high frequencies in visible or ultraviolet, and find out which would be the most significant range for disruption of microtubules. And we can label microtubules, we can see definitely how they respond. With terahertz, we thought this would be a very good frequency to explore, but again, it has a very low penetration depth, so a few microns. We need something that can be delivered to the internal organs. If we could deliver terahertz into the pancreas, for example, and somebody has pancreatic cancer, bingo, this cancer cells will not divide. I can promise you that. So just more systematic, and also engineering, how to deliver maybe by some fiber optical cables or something of this nature, endoscopic techniques.
Okay, thank you. I think that Marcos Vieira has a question. Please go ahead.
Hi, Jack. Thanks for your presentation. I was wondering, if there could be seen a relationship between these results of frequencies and some change in cellular morphology, because we know that there is a relation between morphology and cellular dynamics.
Yes, we have not been exploring that. Of course, it is known that cell morphology is also related to cancer transformation. So if we can reverse the cancer phenotype, the morphology will, and of course that relates to things like endothelial to mesenchymal transformation, right? Because the cancer cells form loose connections, they detach from the tissue and they can migrate, whereas normal cells in the tissue don't. They form a brickwork kind of thing. So yes, I agree with you 100%. That would be maybe one of the directions to pursue to find out if these frequencies also, and that could be very fast, right? Because we can use very good techniques of microscopy, confocal microscopy, for example, or lots of imaging, and do a quick scan of morphological changes. I think that's a very good idea. I never thought about this, so thank you. If there's a patent, you'll be a co-inventor.
Okay, thank you very much.
Great. So I think that we have no more questions. I want to thank Professor Tuzinski for this great talk and for his availability to come here today. Of course, thank you so much. Thank you very much. Thank you. Great talk. Thank you so much. Okay. So what's next? We can close. So how can we do that? Okay. Okay, people, I think that we are closing now. Thank you so much for attending. I'm sorry? Oh, we'll be back in 2 years. Okay, that's new for me. Good. I didn't know that. Great. So we'll be back in 2 years. And I hope that it will be in person. Actually, I expect that we will be able to invite people to come here and to talk directly to us, to interact, and to discuss directly. I hope so. But anyway, it was a great meeting, great talks, fantastic talks. I thank all the speakers for the great presentations. It was very good. Thank you so much.
I got an idea. Ask people to open the camera to make the final picture.
Oh, okay. Can you please open the cameras? I think. I'm sorry.
The microphone.
What's the? No, it's not muted. Okay, so everybody come here. Yeah. Go. Come in. Come on. Yeah. Okay, so. Blurred. Okay, so how can you do that?
Like your ghosts.
I see. But how can we do that?
Sei como é que faz.
Eu sei como é tirar o
blurring. Como é que é o
Eu
blurring?
sei. Desculpa.
Okay.
Tirar filtro, isso?
Desfocar. Não.
Desfocar é o plano de fora.
Okay, that's good now. Okay, so here we are, the organizing committee of this meeting, and we all want to thank you all very much for speaking, for attending, for making such a success of this meeting. Thank you so much. Thank you. And we try to open the cameras, what do you suggest?
Yeah, for people, open.
If you want to, please, if you can open your cameras and then. Okay, so.
Great, Marcelo, Luis.
Here we are.
I'm trying to open my camera, but it's not working. I don't know why.
Okay.
Screenshot.
Oh, Marcelo.
Ok, cadê? Como é que...
Faz um print screen, né?
Okay, on print screen. Okay.
Comando de Alt F4, é isso?
Just a moment. Well, I think that I forgot. How you do a print screen here?
Yeah, we are.
Yeah. What's
Color
the...
4.
Okay, 4, right? So here we are. Smile. That's okay? Go ahead.
Ele fazendo.
Okay.
Esse aí abriu
great.
Existem duas formas. Tem uma que ele copia.
That's it. Yeah, that's
Tem
it.
que fazer, você falar isso.
So, okay, 4.
Da maneira.
And you have to go here. So smile. Thank you. That's great. Thank you so much. Thank you. Thank you all. Thank you. And see you all in 2 years, right?
2 years.
Okay, great. Excellent. 2 years. Bye-bye. Ciao. Bye-bye.
It's already asked.